A warm good morning to everybody present over here, respected dignitaries, respected researchers across the globe. Though I'm saying good morning from India, but I'm sure we have people across the globe over here, so it could be good afternoon and good evening for many. So, this World Fungus Day, that is today on 2nd of October 2022, it gives me immense pleasure to welcome you all to this August gathering. MycoAsia Journal of Modern Mycology, in association with Shri Venkateshwara University, Tirupati, is organizing this fun-filled knowledge gathering, learning, and a day of networking, along with a lot of other activities that we have in store for you, ladies and gentlemen. There are very interesting lectures that we have for you throughout the day. And before we begin to those lectures, let me go ahead and extend a warm welcome to you all, especially Professor Dr. Nalin Wijayawardene from Center of Yunnan Plateau Biological Resources Protection and Utilization, Qijing Normal University, China. Welcome to you, sir. Professor Wijayawardene has been intensive in the field of mycology, ornithology, and has been a field expert in the field of plant ecology as well as conservation biology. So, without taking much time, I would like to hand over this virtual platform to Sir. Nalin, sir, please. Thank you.
Hello everyone. Good morning to all of you. I'm Nalin Wijayawardene, the editor-in-chief of MycoAsia, the Journal of Modern Mycology. So today, on behalf of the organizing committee and MycoAsia team, I would like to welcome Professor K. Raja Reddy, the Honorable Vice-Chancellor of Shri Venkateshwara University, Tirupati, and Professor Chinthala Paramageetham, the head of the Microbiology Department, College of Science, and all the academic members and invited speakers and all the participants to this special lecture series. As you all know, this session is organized by Sri Venkateshwara University and MycoAsia Journal to celebrate World Fungus Day 2022. So we started MycoAsia in 2019, and the founders were Dr. Belle, Dr. Shenoy, Dr. Samantha Karunarathna, Dr. Rajesh Kumar, and Dr. Jayalal, and myself. Our main objective was to promote the sharing of knowledge, information, developments amongst mycology research community, especially in Asia, and to establish a good network between the young mycologists and the eminent mycologists in the world. However, we try to promote, especially in Asian region and especially South Asian and Southeastern East Asian regions. And we could publish our first paper in November 2019. So besides scientific publications, we try to organize online seminars, workshops regularly. Today we have 9 lectures that will be delivered by 9 eminent mycologists, and I hope that participants can gain knowledge the current hot topics in mycology. So without spending more time, I end my welcome speech, and again I would like to thank Sri Venkateshwara University for inviting MycoAsia to organize this wonderful event. And I would like to welcome all of you to this session and hope you will be having a wonderful day ahead. Thank you so much.
Thank you so much, sir, for the wonderful welcome address. Moving forward, we are indeed delighted to have within us today Professor K. Raja Reddy, sir, who is the Honorable Vice-Chancellor of Shri Venkateshwara University, Tirupati. Welcome to you, sir. Under the able leadership of Sir, this university has achieved greater milestones in terms of NAC A grade, university grades, and different QS university rankings. I would like to invite you upon stage, respected VC sir, to kindly deliver your inaugural remarks. Sir, please. Thank you.
Good morning to you, my dear Myco friends. It's a great pleasure to welcome all the delegates and participants to this World Fungus Day Lectures 2022, organized by the Department of Microbiology, SVUniversity and MycoAsia Journal on the occasion of World Fungus Day, which is celebrated on today, that is 2nd October every year. I wish to welcome Dr. Belle Damodara Shenoy, scientist in CSIR, National Institute of Oceanography, MycoAsia, managing editor and other speakers for organizing this event in collaboration with the Department of Microbiology, Shri University, Tirupati. Shri University was established in 1954. This university has been a premier institution of higher learning, research, extension, and consultancy to cater to the educational needs and aspirations of the people, and has played a pivotal role in creating new knowledge, fostering innovation to produce economic value, and addressing societal needs and challenges. Judging by the historical time constants of evolution, SVU has emerged as one of the leading universities in the world in relatively short span of time, only in 7 decades. This rapid stems from the broad recognition that the core activities of SVU have had a demonstrable influence in advancing knowledge that benefit national priorities and global scientific and innovation ecosystem. Our research output is very satisfactory and has crossed the mark of excellence, and it is proved in rankings like MHRD National Institutional Ranking Framework, BRICS World Ranking, QS rankings, and the Asia University ranking. So fungi are crucial to our very existence as a mycorrhizal partners with plants and as major decomposers and nutrients cyclers, not to mention as producers of medicines and food, which they provide to us, to other vertebrates, and to many invertebrates. Most lay people and even many biologists do not, however, realize the importance of fungi. People often think youth in their opinion being colored by sinister connotations from stories and the knowledge that fungi rot our food and homes, kill our plants, and that some are deadly poisons. They do not know that our planet ecosystem would not function without fungi. There would be no fertile soil, no plant life, no herbivores, no carnivores, and no humans, but still the significance of fungi is frequently unappreciated. I'm most happy to be associated with this World Fungus Day. This type of events create awareness on this virtual component of our planet. If people know something about fungi, there is a much stronger chance that they will protect them for the future of our ecosystem and indeed for betterment of mankind. I recall the COVID timings in 2021, two fungi in particular put microbiology on the world stage, making the front page of news outlets globally for the devastating impact on human health. The first, the yeast Candida auris, which was first isolated a few years ago from an ear infection in a Japanese patient, is causing major hospital outbreak globally, showing 5 major diseases. It's seemingly appearing out of nowhere until the first environmental niche was found earlier this year in seaweed off the coast of Andaman Islands in the Indian Ocean. The second disease is mucormycosis, causing large number of invasive fungal infection in COVID-19 patients, with many deaths, especially in India and South America. As such, the unique initiative by MycoAsia to organize the World Fungus Day to promote, raise awareness, and draw attention to fungi global is very timely. MycoAsia microbiology Sri Venkatesh would put on a few additional events in future. Yeah, I'm sure many of you will be looking to pursuing science as a career with passion, the challenges, and derive happiness through providing better quality of life to our people. On this day, let me wish you the future of my country by success in your scientific efforts and missions. My best wishes to all of you. I wish you all to have wonderful festive session in this ongoing event. All the best. Thank you. Thank you very much.
Thank you so much, sir. It was a wonderful, enlightening lecture, and I'm sure we all could relate to the instances that you have drawn from the world of fungi. Thank you so much, sir.
Thank
Moving forward, we also have within us Professor Dr. Chinthala Paramageetham, who is an accomplished microbiologist and who is currently heading the Department of Microbiology at Shri Venkateshwara University, Tirupati. She is well known in the field of research and has significant contribution towards upliftment in the field of microbiology. Let me request you, ma'am, to kindly propose the vote of thanks. Thank you so much.
Thank you, Madam.
Thank you.
Honorable Vice-Chancellor of Sri Venkateshwara University, Professor Raja Reddy sir, respected Dr. Belle Damodara Shena sir, and Professor Nalin Wijayawardene sir, and all other eminent speakers of the day. Faculty from Indian universities and foreign universities, research scholars, students, good morning all. I feel honored and privileged to get the opportunity to propose vote of thanks on this special occasion that is World Fungus Day. I would like to thank all the honorable delegates who blessed us with their presence. Thank you is such a prayer that cannot be seen or touched. It must be felt by heart. Words are not enough to thank MycoAsia managing directors and editors on behalf of the department and university, and our special thanks to Dr. Belle Damodara Shena, Principal Scientist, National Institute of Oceanography, for readily accepting our proposal and lead taken to arrange everything regarding this event, starting from arranging speakers, publicity, to Google platform of 1,000 participants without any assistance from our side. Moreover, he is constantly encouraging microphiles to participate in this big event by releasing teasers and message alerts. Thank you so much, sir. I am also very much thankful to all the invited speakers and readily accepted to give illuminating talks on fungi. On behalf of the organizing committee, convey my deep regard and hearty thanks to our Honorable Vice-Chancellor, Professor Raja Reddy sir, and I am also thankful to the Rector, Professor Srikanth Reddy sir, and Registrar Professor M.D. Hussain sir for giving permission to organize this mega event from our campus. My special thanks to all the faculty friends, research scholars, and students from Indian universities and foreign universities who helped us in spreading the news and also for their participation. Thank you.
Thank you so much, ma'am, for proposing the vote of thanks. And I would like to move on to the next session. So we have two technical sessions divided throughout the day, followed by the valedictory session. So for the participants, I would like to inform you that we have the lectures arranged right one after the other, and they are divided into two main sessions, that is technical session 1 and technical session 2. And we would be beginning with the first session of Professor Nalin. Thank you so much, sir.
Let me formally introduce, good morning all. Let me formally introduce Dr. Nalin. Dr. Nalin is a professor at College of Biological Resources and Food Engineering, Gunjan Normal University, China. And Dr. Nalin has completed his PhD under the legendary mycologist of Marfeling University, Professor Kevin G. Hyde. Uh, his PhD topic was on taxonomy and phylogeny of Demataceous coelomycetes and nomenclature of asexual fungi. And Dr. Nalin is famous for his publication. He has around 130 scientific publications with an H-index of 32. And he's serving as editors for many reputed journals in mycology. And also, we are glad to have him as editor-in-chief for MycoAsia. And further, Nalin is famous among the Asian mycologists for his great publications and contributions towards nomenclature of fungi, especially on the compilation of the word Ascomycota, which is considered to be as a dictionary for Ascomycota, as well as the outline of fungi, which is one of the most cited publications among taxonomy of fungi in the recent past. Let me formally welcome Dr. Nalin. Over to you, Dr. Nalin, please.
Thank you so much for inviting for this wonderful session. So my topic will be the recent changes and important facts in fungal nomenclature. I'm Nalin Wijayawardene. I'm originally from Sri Lanka, but at the moment I'm working in College of Biological Resource and Food Engineering in Beijing Normal University, Yunnan, China. So before we move into the topic, I assume that you all are familiar with common names and scientific names. So briefly, common names are used locally or maybe vary by region or to countries. So, but scientific names, these are unique names used by scientific community to accurately and universally identify a species. Simply, however, we need one name for one organism. It eliminates confusion between different communities or nationalities. Besides, one name is an important in different disciplines, especially in applied sciences such as plant pathology, plant quarantine, plant breeding, and also in clinical sciences. So we know that binomial nomenclature, which was introduced by Carl Linnaeus, is the proper way to name a species. So each and every organism would have one scientific name which would be used by everyone to identify an organism. So that one scientific name has two components, that is generic name and species name. However, in fungal nomenclature, in the nomenclature system, not only one fungal nomenclature, there are different bodies which governs the different nomenclature systems. So International Code of Zoological Nomenclature, or we call it as ICZN, deals with the biological nomenclature of animals. Earlier, the International Code of Botanical Nomenclature, or ICBN, deals with the biological nomenclature of plants, fungi, and algae. So it was established in 1867. However, the term ICBN is no more used. Now we call it as International Code of Nomenclature for Algae, Fungi, and Plants, and it's also called as Shenzhen Code. So when we move into the fungal nomenclature, as I mentioned, now we call it as ICNAFP, and when we are working in rules and codes in nomenclature, there are two important governing bodies like International Mycological Association and International Commission of Taxonomy of Fungi. So when we are dealing with the rules and codes, first inside these community, these association or these bodies, we come to a broad agreement. Then we are going to propose these changes to in the International Code of Nomenclature for algae and fungi and plants. So as I mentioned, it is essential to have one name for one organism, right? So, however, when we move into the biology of fungi, some fungi, they produce asexual morphs and sexual morphs. You all know that asexual morphs is haploid or end stage. And when we talk about the sexual morphs, they have diploid or two end stage. So I'm not going to talk about this too much, but in the right hand side, you can see the morphology, the common morphology of the sexual fungi. So at the same time, this particular species might have an asexual production. So in this case, this sexual fungus also having a asexual morph, and this in this case it's a morph that produce conidiomata or fruiting structure. We call it as pycnidium, and this type of fungi are called as coelomycetes. There is another group of fungi that do not produce closed structures, like in the left side picture, and that group is called as hyphomycetes. So this is the very first report by Tulsane and Tulsane as a drawing of both sexual and asexual morph of powdery mildews. That's meaning in class Leotiomycetes. So in the top picture you can see the sexual morph, while in the bottom picture you can see the asexual morph or the anamorph. However, then we need to understand there could be the term the whole fungus that which comprise sexual morph and asexual morph. So in that case, the whole fungus is called as whole morph, and sexual morph is also called as teleomorph, while asexual morph is called as anamorph. You can use any of these terms, and in some cases, some scientists argue that whole fungus, when we're talking about the whole fungus, some species they do not have an asexual morph. So in that case, whole fungus would be sexual. In the same way, in some cases, whole fungus could be the asexual morph, that is, they are lacking any sexual morphs. However, in some cases, some fungi, they do have more than one asexual morph. So in that case, we name them as syn-asexual morphs or syn-anamorphs. These are some important terms when we are talking about taxonomy and ultimately in fungal nomenclature, right? So we believe at least 20% of the species show more than one morph in their life cycle. That is, they are pleomorphic species. And when Kendrick 1979, he emphasized it is not they occur as sexual and asexual morphs through their life history, but may be separated in time and space. Okay? However, these morphological characters of these morphs are quite distinct. In case of sexual morph fungi, they produce ascomata, ascus, and ascuspores, while asexual morphs produce conidiomata. In some cases, they do not produce conidiomata, and conidiophores, conidiogenous cells, and conidia. So I would like to recall this particular slide again. You can see right hand side with the ascus, it's easy to identify the sexual fungi. On the other hand side, the other species, they do not produce ascomata or ascus. So those are called as asexual fungi, but in this case, it is called as coelomycetes. So we have two types of morphological groups in our environment. In this case, to name those species, Saccardo introduced dual nomenclature system of fungi. So asexual fungi and sexual fungi, they have different names. And this dual nomenclature system was accepted by the International Botanical Congress in Vienna in 1905. So simply, when one fungus has two morphs, sexual morph has one name, while asexual morph has another name. Right? In this case, this species, let's see, Cochleobolus, this particular it has a different morphology. So if we talk about the generic level, so it has a separate generic name, while it has a separate generic name for its asexual morph. So this is another example, the Communitaspora in 1996, introduced by Ramale, Dr. Ramale. She found the sexual morph from the environment, and she got the PO culture, and the asexual morph was correlated in the Pio culture, and she has to name it Hypospora, although it's from the Pio culture of sexual morph, because we have to follow the rules according to the international code. And the Hypoxylon is another example. In the same case, they have found the sexual morph, and they found the Pio. They have the Pio culture, and the Pio culture, in the Pio culture, it produces the sexual morph. So they have to follow the code, and they have to give two names for the one fungus. Right? So you can see how it will be complicated. Right? So how can we link these different morphs? So in traditional approach, two morphs at the same time on the same host. So we call it as co-occurrence on the same host. On the other hand, we can use culture-based methods, as I mentioned in the previous slide. So in most cases, they produce the asexual morphs in the single ascospore isolates. However, now in these days, we use DNA sequences linking these different morphs. So I already told you that it is not necessary to have both morphs in the same host, on the same host. Right? Maybe you can find an asexual morph from the environment, but earlier the sexual morph could have been published or described by some other person. But in this case, we can easily link sexual and asexual morph based on DNA sequences, because genetic material will be identical. So in this case, this is another good example for this pleomorphism. So you can see the Botryosferia dothidiae, well-known fungus from family Botryosferaceae, the sexual morph, the left corner slide. You can see the middle one, we call it was named as Fusicoccum, and the right-hand side you can see Dichomera. Right? So in different time, different people introduce these generic names, but in the when we are going to the DNA sequence based phylogenetic analysis, they group together in phylogenetic analysis. So simply these all different morphs representing one group of one genus or one species in some cases, right? So this is how we show it in one of our papers. You can see it Dichomera. It groups with the Botryosferia coccus and Botryosferia sensu stricta. So this, when we talk about the generic level, so we have linked different sexual morphs with their asexual morphs based on the phylogeny. So these are two examples. Wichnadia you can see in the left side. Wichnadia is the sexual morph. The asexual morph is it's asexual. Phyllosticta is its asexual morph, and you can see the right-hand slide. You can see the asexual morph is Stemphilia, and the sexual morph is Luvia. Right? So then why we can confirm that these morphologies are belonging to the one species, or one, maybe if we talk about generic level, to one genus. So why we are using different names? So then scientists, it was a long-term discussion, and then in 2011 we tried to propose one name for one fungus. Right? So then there's a huge debate which name would be accepted. So there should be some widely used names, especially when we're talking about the pathology. But in some cases, we have to use older names. However, there is a broad agreement now. We are going to use the older name in generic level. So this is again the same slide. So we have Wichnadia and Phyllosticta. So Phyllosticta is the asexually typified name, but it's the older name. Now we are using the older name, older generic name, that's mean Phyllosticta over Wichnadia. So all the Wichnadia species name have to be transferred to the generic name Phyllosticta. Then Stemphelium and Luvia, again, the asexual typified name Stemphelium was accepted over Luvia, the younger sexual typified name. In this case also, as I told, we accepted asexual name over the sexual name. Because I emphasize this because earlier there was a rule, if we are going to use the asexual name over the sexual name, they had to get a special, how to say, we had to do a proposal. But now we don't need to do it. All the names, or whether it's sexual or asexual, they will be given the same priority. This is another example, Ramularia versus Mycosphaerella. It was also well established. It has very good established links. So right hand side you can see the Mycosphaerella, the sexual morph, and left hand side you can see Ramularia, the asexual morph. So it was proposed to adapt Ramularia over sexual name, that is Mycosphaerella. So in some cases, when we are going to this species level, this is another interesting story, that Paraphyosferia mytochi. It was the left-hand side one. It was introduced in 1967, and in 2018, Vanasinghe et al. Found the both species from Rosa species. And you can see they have distinct morphologies, but in the genetic level they are identical. Therefore, Vanasinghe et al. Did not propose a new generic name for the Coelomycetes asexual morph, because genetically they are identical. So they have proposed the asexual morph of Paraphyosferia mytochi. So that is why we need to use phylogenetic analysis or DNA sequences in the current mycology. This is also the same story, but in recently planned in 2018, they have found this above sexual morph from the host locus, and they have got the cultures of the ascospores, and then that cultures correlated the asexual morph, and you can see the bottom picture. So they have, although it's the different morphologies, based on new nomenclature, we have to give only one name. So they have named it as Dendrostroma coccinum. So this is an interesting story from the same publication in Fan 2018. They have found different asexual morphs. In the above picture, you can see it's a hyphomycetes morph, and the bottom you can see asexual coelomycete morph. And although they have different morphologies, in their phylogenetic analysis, they confirm both morphologies share identical DNA. So they proposed only one name, that is Cinemasporella aculeus. So, but it's not necessary to have the both morphs at the same time, right? So in this in same genus, Cinemasporella toxicadaria, they produce only the hyphomycetes morph. So they have given a different species name, but under the same genus. Right? So this is the current status of our mycology. I mean, regarding the nomenclature. So especially when we are talking about the generic level, it seems we are trying to dealing with the generic level since 2011, 12, 13, and now it was high. There are a lot of debates among the communities, and now it little bit stabilized. We have to say. So there are about 8 to 10 publications have been published by different groups, different research groups, and for targeting different classes or orders. So these are some examples, and it's very important to have a broad agreement when we change do the name changes. So it is important to have a very stabilized nomenclature in the future. So besides these nomenclature changes, we have to understand some other ground rules when we are naming a fungus. We have to designate the holotype. That means the specimen that particular species was described. So we have to designate the holotype. If it's a new species, we have to designate the holotype and we have to indicate where it will be stored. If you don't use these, if you don't mention this thing in your publication, the names will be invalid. Right? So earlier I told that at the moment the naming the algae, fungi, and plants are governed by one nomenclature system. So we have to check whether that one generic name was used in previous cases. I mean, if we want to use one name for our fungal new genus, we have to check that was used by whether it was used by some plant botanist or some algal experts. If we use the same name again, then your name will be illegitimate, right? So then we have to obtain the identifiers from accepted repositories such as Index Fungorum, Mycobank, or Fungal names. And when we are proposing new combinations, that means when we want to transfer one genus to another genus, we have to check the very first name of the taxon. That's mean the best unit. Otherwise, again, your name will be invalid. So these are when we are working on huge publication like Outline, these type of mistakes that came across always. So I thought to give these when you are working on taxonomy, these and nomenclature, these are very important, right? Then we have to think about the future of fungal nomenclature. So as I told, ICNAF still requires a fungal names to have a type. So it could be a specimen or a specimen, or alternatively we can have cultures, or in some cases we can have illustrations. Right? But we all know that it is difficult to get cultures of some taxa, and in some cases they do not produce morphological structures. Therefore, now ecologists, they have used high throughput sequencing or next generation sequencing, and they reveal a lot of novel taxa as well as new lineages in kingdom fungi. Hence, now taxonomists and mycologists discuss the possibility of new nomenclature which is based on only the sequences. However, this is still a volatile topic among the community. Hopefully, in next International Mycological Congress in 2024, we can come to a conclusion about this topic as well. So this is the publication that recently published about this sequence-based nomenclature. But still, I have to say that although even though in some authors are here, they have some different opinions. In my case, I should say I support this proposal. So this is the end of my presentation, and I would like to acknowledge Dr. Volker, the curator of Index of Fungorum. Then I would like to thank Dr. Belle and organizing committee. Thank you so much. In any case, you can contact me from my email and my research and Facebook, and my research interests are taxonomy and phylogeny of asexual fungi, timber-associated fungi, and especially in fungal nomenclature. Thank you so much for your attention.
Thank you, Nalin, for a wonderful deliberation on fungal nomenclature and its updates. The last 10 years were in a mess for considering the fungal nomenclature and the implications, considering the drastic changes which were happening in the modern mycology. And we are happy that we listened most of the key points from you. Now I would like to open the session for questions. If audience, you have any questions, please.
Sir, I have a question.
Yes, Yogesh, yeah.
Sir, for example, what you said that for a valid publication that a species should be registered in Mycobank or some in registration societies like Traces of Fungi, Mycobank. Now, my question is that, for example, a person who has recently described a species, he has not registered his species in mycobank basis of fungi, so that name is invalid.
Yes.
If
Yes.
that name is invalid, and for example, I am proposing a new species, I am describing a new species, so whether I have to cite his paper about that species that he has not made a valid name, and if it is not valid, then what I should write it as Norman illegitimate name or some other thing.
Yeah, I think Yogesh, you have to cite. We have to give the courtesy for that particular person, but you have to mention it is invalid, and you have to mention what is the reason. Because in
Okay.
some cases, some taxonomies they're doing some mistakes. They register their name index fungorum and at the same time in mycobank. It is not necessary, but this
Okay.
is another case, right? So I know that in every week they synchronize the names, right? If you register it mycobank, that will be it. So if you don't register, then uh, if you don't register it in any repository, then it will be surely invalid, right? So
Okay.
then, but later but if I come across that type of case, personally I write to that person who described that particular species. Then they can contact Index Fungorum or Mycobank. They can write a small validation to Index Fungorum, right? So I think
Okay.
therefore you have to mention about that particular name. In my case, if I normally do it, although it's
Okay.
invalid, right? And
So
if I want to compare the morphology, still I'm doing it.
okay. So if I
Yeah.
am mentioning that species as an invalid name, then in that case I have to write it as Norman illegitimate in that context, or what?
No.
No.
Actually, there's two cases. So that's what I mentioned. When the genus name was taken earlier by a plant name, and you are going to use the same name for a fungal in generic case, right? In generic
Yeah.
level, then your
Mhm.
name will be illegitimate,
Okay.
right? If you don't
Okay.
designate a holotype, then it will be invalid.
Invalid.
There
Okay.
are two
Okay.
cases
Okay.
because these things
Thank you.
are governed by different acts in the code. As I remember,
Yeah.
if it want to be invalid, it's under Article 43.1, as I remember. But if it's
Yes, sir.
illegitimate, it's under Article 59.1. It's called as then now it's called as I can't remember exactly. It's a separate section for fungi. So it's two
Fungi.
different
It's a
cases. Still you can validate them. Hmm.
okay. Okay.
Yeah.
So nice, of sir. Thank you very much. Thank you.
Thank you, Rajesh. Thanks.
Thank you, Nalin. Shailesh, you have any question?
Hello. Can you hear me, sir?
Yes.
Yes, sir. So you mentioned regarding something about sequence-based taxonomic identification, sir. I'm a very novice in fungal taxonomy, but I frequently work with sequences, and I do DNA barcoding of pteridophytes and low vascular plants and other things. Can I use DNA barcoding in fungal identification? And if yes, what kind of sequences are valid and will give me significant data which will help me identify different kinds of funguses? Because I do not have much background in classical taxonomy and fungus.
Yeah, actually, I think in next few lectures you can get a good knowledge about this. In my case, I'm going to talk about only the nomenclature stuff. But you know that currently we believe that IPS region is the best as the fungal barcode or universal barcode. But there are some arguments for that as well. But I think you can you can learn a lot from Dr. Shenoy's lecture and maybe from Dr. Rajesh's lecture also. It's better you talk with them. I'm not going to talk about the taxonomy level at the moment, right? But there are some, especially
Dr. Sridhar Pichai.
Yeah, Rajesh, as I'm correct, right? So we have some sessions to talk about the sequences in next lectures.
Yes, Nalin.
Yeah,
We can discuss such matters in the other lectures.
other lectures. Yeah.
Dr. Sridhar Pichai, you have raised your hands for question.
Hello, sir. Hello.
Yes, please go
Yes,
ahead.
yes. Yeah, please.
Sir, good morning, sir.
Good morning.
Sir, I got the sequence and ITS and LRO region, both of them in one. This is called in dichondreless. But that is earlier is described in only in the microscopically. Right now, I sequence in both of them in microscopically, and microscopically is matched to the dichondreless. Earlier person is not deposited into the sequence in the NCBI. Right now, how do I to deposit the genus in newly in NCBI? Hello, sir.
Yeah, I didn't clearly. I am not very convinced with the question. But you're talking about that particular species have the sequences, right? But it was not deposited in the gene bank.
Yes, earlier worker is not deposited. Right now, I got that culture and I will sequence in IDS and LRO regions. I have to is deposit in newly in that genus into in NCBI.
I think there is a process for to deposit them in the in the gene bank, right? But
Yes.
if you have any question regarding the nomenclature, I mean, how the importance of the when what are the necessities to establish a new name, I can help you.
Okay.
But in this case, I think it's a not long process, but it's a different topic.
Okay, sir.
Like there's a process to deposit your sequences in the gen bank.
Not
Right,
deposited, sir.
like
That is okay.
Yeah.
Dr. Sridhar, you can clarify these questions later on with the experts panel of MycoAsia. We are happy to help you. Let's continue with further questions from audience.
Hello.
Yes.
Yeah. Hi, sir. To
Yeah.
propose as a new species, do we have to submit that holotype to any kind of herbaria, like national or international herbaria, right?
Yeah, it is essential. Yes.
Oh,
And you have to mention
what happens? Yeah, what happens in the few cases when we go to the field, we'll get hardly 2 or 3 fruiting bodies that will used for the identification for the molecular. That time, what we can do, like we don't have enough bodies to submit. We have already used that for microscopy, for ITS sequencing, for cultural studies.
So you can, if you
is
have,
it
if you have the culture, you can preserve it, right? Or maybe you can,
okay?
yeah, it is also will be fine. But at the same time, you can try to get the how to say, the fruiting structures on the culture. You can do some techniques. You can try to induce the cultures to have the fruiting structures from get the cultures fruiting structures from the culture. That is also possible. And we have one technique to use dry cultures to maintain as the herbarium specimens. Right? It is also valid. It's not a problem, and that's why
Okay.
yeah. On the other hand, you can how to say, illustrate your species very well, and you have the cultures, and you can use the culture as the X type, and at the same time you can use the dry cultures as the herbarium specimen.
Yeah. Thank you so much. That is a great help. Thank you.
No, thank you.
Thank you, Sushant. Any more questions? Anyone from MycoAsia have any questions? Especially Nalin has, we should congratulate Nalin for being a part of International Commission for Taxonomy of Fungi, because he is a elite panel member now, and we are delighted to mention this point. I missed it during the introduction.
Excuse me. Hello.
Yes, sir. Go ahead.
Hello.
Hello, sir.
This is Arun Karwar. from BHU. Hello, Nalin.
Yeah,
Hello.
I can hear you, sir. I can hear you, sir.
Yes. Can we include secretome also while designating the name of any fungus?
I didn't hear
Secretome data.
I
The
didn't hear the
data,
first part.
as the polyphagic approach is being adopted,
Yes,
so
yes.
should we also include secretome data while designating the name?
You mean genomic data? Yes, we have to.
Means metabolic profiling should also be included.
Yeah, in some cases, if you have some data, especially the taxa from Sileria and in some lichen taxonomy they use, but I don't have much experience on metabolic profiling. But I normally use morphological characters and DNA sequences. That because you know that we don't have for all species, we don't have DNA sequences even for DNA sequence for all the species at the moment. So I think it's not not a good idea to use metabolic profile for at this moment. But for some groups, yes, you can use. Yes, you can use.
So, how many? I mean, what will you suggest to our youngster that whenever they are going to describe any new novel species, so how many genes they should include, and what are those which are really essential? So they should focus to those only. Otherwise,
Yes.
they include many which are really not required. So this is the forum where from this message could be given to the scientist or youngster, especially for the clarity.
Yeah, normally in my case, I normally use ITS and elongate factor, RPB2, and LSU and SSU for high level ranking. So, but it depends. If you are working on some Botryospheriae, they normally we use. So it depends, and if you can compare the morphological characters first with the other important publications, then you can pick what are the necessary or what are the important barcodes for that particular group. In my case, I normally I normally do the sequence for ITS and LSU. Then we can easily get the closer street from the gen bank, and then according to that we select the important the other important barcoding and other barcodes. That's the way I'm doing. So normally, as I'm closely working with Professor Kevin, you always need at least 5 genes: LSU, ITS, SSU, RPB2, EF. Those are very important. So if you can target on those regions or loci, I think you can do a good job.
Okay, thank you, Nalin. I think it will be clear to other people also. And thank you for delivering your nice lecture.
Yeah, thank you so much, sir.
Congratulations.
Thank you,
sir. Thank you for asking a wonderful question, sir.
Yeah.
With this question, let's conclude this session. We genuinely thank Dr. Nalin for his talk, and let us move ahead with the next talk of Professor Samantha. Let me formally introduce Professor Samantha. Professor Samantha is a part of Center of Union Plateau Biological Resource Protection and Utilization, College of Biological Resources and Food Engineering at Gunjan Normal University. Professor Samantha is known as a face for mushroom in China and across Asia, and he has published around more than 200 SCI publications, having an H-index of 35, and he is one of the most cited young scientists from Asia, having 6,500 citations, and he is an integral part of core group of MycoAsia. On behalf of MycoAsia and the audience, we we welcome you on the platform. Please, it's over to you, Dr. Sam. Thank you.
Yeah, thank you very much, Dr. Rajesh, for introducing me. So first of all, happy World Fungus Day to you all who are joining from all around the world. Good morning, good afternoon, good evening to you all. So my topic today is mushroom poisoning in Asia. This is a very interesting topic and also also a topic that we should pay much more attention to. So let me start with why we collect wild mushrooms. So as you all know, there are so many reasons why people collect wild mushrooms. So one reason is people like to go into forest and take some photographs of mushrooms. And some people like to go into forest and see these beautiful mushrooms. And of course, some people, they like to go and collect wild mushrooms and for the purpose of food and for purpose of medicines and some other scientific reasons. So as I mentioned, there are different reasons why people collect mushrooms. So one, because of the scientific interest, and also some people like to take nice photographs of mushrooms, and some people like to collect mushrooms for food and medicine. So the most important thing is when you collect mushroom and when you are in the forest, you should be careful because not all the mushrooms are safe. Some mushrooms are extremely dangerous. So this is very important. So next slide, please. So let's see how serious is this mushroom poisoning in the world. So the estimation is at least about 100 people die every year worldwide, but this number might be underestimated given the fact that about 50 to 100 separate deaths report in Europe and China. So the number should be at least 200 or more. Next slide, please. So let me take an example, Yunnan province of China. So if I talk about the mushroom poisoning in this Yunnan province, so every year there are a lot of mushroom poisoning cases reported in this province because people are fond of wild mushrooms. So here there is an interesting fact that in 2020, there were 190 cases of mushroom poisoning, and there were 2020 there were 81 incidents and 12 deaths reported because of mushroom poisoning. But if you consider the COVID-19 virus, only 2 people died because of COVID. So you can see the mushroom poisoning is serious compared to other places in the world. Next slide, please. And when we see the whole picture of mushroom poisoning in China, you can see from 2019 to 2021, In 2021, only 20 deaths were reported, and in 2019, it's 25, and 2020, it's 22. So the good news is, in 2021, it's less than 2019 and also 2020. So this is a good thing that we see the mushroom poisoning cases reduce year by year. Next slide, please. So let's see why there are mushroom poisoning cases in the world. What are the reasons? So one is insufficient knowledge and unclear taxonomies and increased consumption and misleading folklore. So in this point, I have to mention that there is no universal test to identify poisonous and edible mushrooms. So we have to be very careful. Next slide, please. So recently there were some papers published on identification techniques and detection methods of edible fungal species. So this is one of the papers that was published in Food Chemistry. So in this paper also, it was mentioned that there is no universal test, even though there are some localized tests for different mushrooms or different genera, but there is no universal test to see whether there's a mushroom is poisonous or not. Next slide, please. So in mushroom identification, spore print is very important. So spore print tells you the color of the spores of a particular mushroom, and depending on different genera, the spore color are different. So for example, Amanita has white spore prints, Wolvariella has pink, and Chlorophyllum has olive green, and Agaricus has chocolate brown. So spore print is very important in mushroom identification. Next slide, please. So one reason for mushroom poisoning is the look-alike mushroom. That means some edible mushrooms look very similar to poisonous ones. So there is one example here, Macrolepiota procera, which is an edible species, and Chlorophyllum molybditis is a poisonous one. So you can see by looking at the morphology, it's very hard to distinguish these two. But if you look at the underside, Macrolepiota procera has white gills, white spores, and Chlorophyllum molybditis has olive green spores or olive green gills. So by this character, you can positively distinguish these two. Next slide, please. So be aware of these poisonous and edible mushrooms, because otherwise you get deadly mistakes. So there's another example here, the dead cap when it is young, and the paddy straw mushroom. You can see they look similar, and especially people from tropical areas, when they go to temperate areas, they mistakenly collect this deadly poisonous dead cap by thinking of this paddy straw mushroom. So it is very important. You should understand different regions, they have their own mushrooms. So don't think or don't expect the same mushrooms in other areas. This is very important. Next slide, please. So some mushrooms are delicious, some are deadly. So depending on the nature of the mushroom, whether you pick it or you ignore it. So here also the Wolverella wolvacea and the Amanita virosa, the deadly poisons. Next slide, please. So again, you can see Wolverella wolvacea and the Amanita phalloides. They look very similar in morphology. Next slide, please. And here you can see the edible Mochella and the poisonous Gyromitra. So the morphology is very similar. Next slide, please. So here you can see the black moral, white moral, and the false moral or the Gyromitra. So you should be very careful not to be too excited and collect the wrong ones. Next slide, please. So here another example, Lepista nuda, it's edible, and Cortinarius archerii, it's poisonous. So in Cortinarius, there is a brown cortina on the stipe, but in Lepista nuda you don't see it. So this is one of the positive characters you can distinguish these two. Next slide, please. So again, this is Lepista nuda and the Cortinarius. You can see the cortina on the stipe. Next slide, please. So this is another example, Pluteus and the Entoloma. So when you see the morphology, it's very similar. So should be careful by distinguishing these edible and poisonous ones. Next one, please. So we can see very often there are headline news regarding the mushroom foraging and mushroom poisoning. So people are always advised when they go into forest, be careful about mushroom that are poisonous. But still, people get poisoned because of some people. They are too excited when they see mushrooms, and the other thing is people are fond of wild mushrooms in some regions. Next slide, please. So these are some case studies that were reported about deadly mushrooms. So this particular mushroom, Trogia venata, this was reported in Yunnan province, and after several people have been killed by this mushroom, this was identified as a deadly mushroom. Next slide, please. And these are also some of the mushroom poisoning cases around the world. Iran and other mushroom poisoning cases around the world. Next slide, please. And this is a case from Jordan and also from China. Next slide, please. So recently, we saw a very touching news that was in Assam. So this case was reported in Assam. So some people who were working in a tea garden, so they were killed by some deadly poisonous mushrooms. And in that report, I saw the main reason was the lack of awareness of edible and poisonous mushrooms. So it was stressed that people should be educated, or people should be known about these edible and poisonous mushrooms. Next slide, please. And at the same time, I saw this paper was also published in Toxicon by some Indian authors. So in this paper also, they mentioned the main reason for mushroom poisoning in India is the lack of the awareness of poisonous and edible mushrooms. So this is reason that Indian mycologists should more focus on this particular topic and train the locals or people who like to collect wild mushrooms how to identify these poisonous and edible mushrooms. Next slide, please. So these are some of the misunderstandings regarding wild mushrooms. So some people think if the mushroom peels well, you can eat it. So it is absolutely not correct. And some people believe all mushrooms growing on wood are edible. That is also not true. Mushrooms that squirrels or other animals eat are safe for humans. That is also absolutely not true. All mushrooms in meadows and pastures are safe to eat. That is also wrong. All white mushrooms are safe. That is completely wrong. So some of the deadly mushrooms are white, and poisonous mushroom can be detoxified or by boiling or drying or pickling. That is not true because the alpha amanitin in metaphalloides cannot be destroyed by heat. And poisonous mushroom tarnish silver spoon. That is also completely wrong. Next slide, please. So these are some of the regional misunderstanding folklores. So in Myanmar, people think all red mushrooms are poisonous and all white mushrooms are safe. So this is completely wrong. As you all know, Amanita virosa is white, but it is deadly poisonous. And in Laos, people think if you boil the mushroom with cooked rice, if the mushroom changes its color to dark blue or black, then those mushrooms are poisonous. If the color doesn't change, those mushrooms are edible. So this is true for some specific mushrooms, because the iodine test, if the mushroom contain iodine, this is like a starch, the test. So this is true for some particular mushrooms that contain iodine, but this is not true for all the mushrooms. And in Sri Lanka, people believe that a poisonous mushroom always should have a ring and vulva. You can see the mushroom in this photo, Tricoloma conudame. It's a deadly poisonous mushroom, but you don't see any cap or stipe. It's like a coral. Next slide, please. So these are some of the recommendations to avoid mushroom poisoning. So don't eat unfamiliar species. Examine each and every specimen when you collect mushroom. And keep your collections separate. Don't mix your collections together. And do not eat raw wild mushrooms. Always try to cook your collection, even though some mushrooms are known to be edible as raw. But it is always advised to cook your mushrooms. And only eat good specimens. Don't eat the mushrooms that are damaged by some things or eaten by some animals. And don't try to collect mushrooms from contaminated habitats. As you know, mycelia absorb these secondary metabolites, the poisonous, the chemicals in the soil. like pesticides, insecticides, and heavy metals can be absorbed from the soil. So don't collect mushrooms from the contaminated habitats, and keep your collections in a cool, dry place. Otherwise, the mushroom go bad very quickly. And don't try to experiment with mushrooms. If you don't know a mushroom very well, don't eat it. And there are some specific mushrooms, such as Coprinus comatus. If you eat and drink alcohol, then there it will be a reaction. So it is not a pleasant reaction. So you should not consume these kind of mushrooms and drink alcohol. And if you have an extreme fear to eat mushroom, don't eat. And don't give mushrooms to susceptible people like young children or very old people, because it is very difficult to digest mushroom for people if you give them mushrooms. And also don't be greedy. Anything too much is also not good. Next slide, please. And when we talk about mushroom identification or the local knowledge, we cannot ignore, because people take this knowledge generation to generation. So it is important we talk to local people and understand what they know about wild mushrooms, and try to link this knowledge into the scientific knowledge, and then try to understand why these people tell this kind of things. This is the scientific reason, and if you combine this together, you can get a good identification about your wild mushroom. Next slide, please. So when we were doing some work in Myanmar, we understood that their wild mushroom knowledge was very poor, and that's why they had some beliefs or the folklores regarding these wild mushrooms. So when we were there, we tried to train them to collect their wild mushrooms and identify which ones are edible and which ones are poisonous. Next slide, please. So, and also we prepared some posters for them to identify their edibles and poisonous mushrooms in their forest. So these kind of things are very important for the local people to understand which kind of mushrooms are edible and which kind of mushrooms are medicinal, or which kind of mushrooms are poisonous in their region. Next slide, please. And these are some of the photos that we took during the mushroom trainings we had in Myanmar. So we went out to the forest with the local people in Chin State, and we collected mushrooms together, and then we came back and we spread the mushrooms, and then we tried to identify them together, and it was very successful. And I'm sure now they are using their forest resources properly, because now they have an idea which ones are edible, which ones are poisons. Next slide, please. And these are some of the observations I had, especially in Yunnan province. So I often see these kind of posters are displayed in restaurants and public places to show people which mushrooms are poisonous and which ones are edible. But I want to stress that if you want to produce a poster to to let people know which ones are poisonous and which ones are. So it is better to include as much as possible characters. Otherwise, people are not able to identify these mushrooms properly. I suggest you can include both upper side and lower side and all the colors in a high resolution mode, so that people can positively identify what are these mushrooms. Next slide, please. And this is another observation I had. Like, this paper was published on mushroom toxicity recognition based on multi-graded cascade forest. So the authors used a computer program to identify the toxic mushrooms. So I particularly, personally, I don't like this kind of identification techniques, because you cannot input each and every character of mushroom to a computer program to identify mushrooms. This gives a wrong impression for the people that only these characters are the ones that are known for poisonous mushrooms or for edible mushroom. So this is completely a wrong way of identifying mushrooms. Next slide, please. Yeah, this is the details of the computer program that they use for mushroom identification. You can see they use only some features of mushrooms. So this is completely not correct. Next slide, please. And this is another study I saw, and here they use the molecular methods to identify mushrooms. So this is a good way of identifying, because it is reliable and it can give good results. Next slide, please. Yeah, I think that is the end of my presentation. So thank you very much for listening. And my take-home message is, never eat a wild mushroom if you don't know 100% about that mushroom. And I would like to take this opportunity to thank the organizing committee and Dr. Shenoy and MycoAsia for organizing this event. And thank you very much. And if you have any question, now it's time for questions. Thank you.
Thank you, Sam, for a wonderful session on behalf of MycoAsia. Now we invite your valuable questions, please.
Can an edible mushroom or a non-poisonous mushroom grow in the same media side by side along with a poisonous mushroom?
Yes, that's why I specifically mentioned that we should be careful about these lookalikes. Sometimes the edible one and the poisonous one, they look very similar. So they grow in the same area. So people often confuse. They think this is the edible mushroom, but they collect the lookalike poisonous one. So this is very common reason why people get poisoned by mushrooms. So it is possible to grow the same edible or poisonous in the same area.
So, sir, what is the factor which is making the one mushroom poisonous and the other mushroom edible, being in the same environment or the same substrate?
Yeah, this is a very interesting question. Still, people have not really understood why. because we can see in the same genus sometimes some are edible, some are poisonous, and some are even deadly poisonous. So evolutionary, maybe it's a different story that when mushrooms evolve, in one point, some mushrooms became poisonous and some mushrooms became edible. But the puzzle is why in the same genus some are edible, some are poisonous, and so there's no pattern you can see in this poisonous or edible mushroom. So it is a very good study if someone can answer these questions. Why some mushrooms are edible and some are poisonous, even in the same genus. So this is still a puzzle. Nobody has really understood.
Thank you, sir.
Dr. Hashma, you have any question? Dr. Sridhar Pichai, you have raised your hands for question. So Jackson, please.
Sir, thank you for the wonderful presentation. But I'm just a student who is new into mycology. But you told in your slide that there is no universal test for poisonous mushroom identification. So, sir, does you imply that it is impossible to make a universal test, or can it be possible in the near future?
I should say, like, for my thinking, I feel it is impossible to make a universal test because mushrooms contain so many different kinds of toxins, and this is the reason why the scientists cannot make a universal test. Because if you specify some chemicals or the toxins and make one universal test, and if somebody finds a new poisonous mushroom, and if the person finds a different new toxin, then the universal test become useless. So this is the reason why scientists are struggling to make a universal test for mushrooms. So for my understanding, it will be impossible to make such a test. That is why there are localized tests for different genera of mushrooms. For example, in agaricus, we have one test called Schaeffer's reaction. So if you take phenol and concentrated nitric acid, and if you make one line on the cap of agaricus from phenol, and if you cross the line with concentrated nitric, so if the crossing point, if you see a reaction with a dark red spot, so that is called Schaeffer positive. So that means that mushroom is edible. So if you see Schaeffer negative, so you say that mushroom is not edible. So these kind of localized tests are available, but there's no universal test at the moment because of this variety of toxins in mushrooms.
Sir, can I please follow up?
Yes.
Professor Atri, sir, do you have any questions?
Thank you, sir. Thank you, Dr. Rajesh. I don't have any question. I just wish to congratulate Dr. Samantha for the excellent presentation and for clearing all the myths which the mushroom poisoning is, with which it is associated. He is very right in saying that there is hardly any test known so far, and it is not possible in future as well because of the type of variety we have in mushrooms and the diversity of toxins within the same genus. Even see Amanita, there are edible species, there are poisonous species within the same genus. There are a number of such examples, and excellent presentations, sir. Congratulations.
Thank you very much.
So, sir, can I please continue one more?
Professor Sharma, you can go ahead. Jackson, you may please wait for.
Yeah. Dr. Samantha, it's a wonderful presentation. What I see is a surprise addition of one trichoderma species in the poisonous fungi, because basically trichodermas are saprophytes and harmless. I see one trichoderma species also listed there.
Yeah, this was reported in Australia, Japan, and these kind of countries as a deadly poisonous mushroom, and I was also surprised to see this is a deadly poisonous mushroom. So you can see this is why we are puzzled about this edible and poisonous mushroom. You never know when you find a poisonous or deadly poisonous mushroom in a genus of maybe well-known edible mushroom. So this is still a puzzle scientists are trying to understand. Thank you.
Jackson.
Thank you, sir. So, sir, these poisons are just like are normally secondary metabolites and have certain biosynthetic pathways inside the mushroom. So, sir, if we somehow identify the genes, suppose in a particular genera, like which genes are responsible for which toxins or poisons that are produced, can't we make a catalog of the particular genera so that we can identify mushrooms that are poisonous or non-poisonous?
Yeah, so this is the way to go. Like, you should pick up the genera wise the mushrooms, and then you can make tests for each genera, looking at their toxins that they produce or the secondary metabolites. So this is I think possible, but looking at a universal test, it is impossible. So what you say is correct. We can make a test for genera wise. That is a good suggestion.
Thank you, sir. And one last thing, sir, if I'm not. So suppose, sir, if I somehow find a noble mushroom and then that it belongs to a particular genera, which are all edible. So, sir, how can I test that the novel mushroom is like edible or non-edible?
Yeah, so if you want to know a mushroom is edible or not, so the way is to analyze the secondary metabolites or the toxins present in the mushroom. So you should go from the beginning, looking at the known toxins. You should analyze these toxins in the mushroom, and also you should look at whether there are heavy metals. So you should go step by step to find out whether these mushrooms are edible or poisonous. So it's not a simple thing. It's complex. You should do all these steps to find out whether it is edible or not.
Thank you so much, sir. Thank you.
Any more questions from audience? Any more question from Hashma, Sridhar?
Am I audible?
Yes, please go ahead.
So I have
Yes, please go ahead.
Eating mushrooms with fish or meat safe or not? Eating mushrooms?
Yeah. Eating mushroom with meat or fish, that is totally fine. But as I mentioned, there are some specific cases, like if you eat some mushrooms with alcohol, then there are some reactions like Coprinus comatus. So apart from that, eating mushrooms with meat or fish, totally fine. Because in Yunnan province where I work, there's a famous dish called mushroom hot pot. So they boil different mushrooms with meat in one pot. So this is really a nice kind of food here. So I should say it's totally fine.
Thank you, sir. My second question is,
Sir, I got one doubt, sir.
pardon. Sir, can I
Please wait for,
continue?
Yeah, Sarala, please continue. Yeah.
Okay, sir. Are all iodine containing mushrooms are poisonous?
So if the mushroom contains too much iodine, so this is really not good, because as I mentioned, like anything too much is not good. When there are standards for certain food, so we say that if this heavy metal or this chemical present above this level, it is poisonous. So that means even iodine, if it is present in mushroom in a certain level that exceed the safe level, so it is poisonous. So that's why in one folklore I mentioned that some locals in Laos, they believe that if they cook mushroom with the cooked rice, if it turns to dark blue or black, so they believe it's poisonous because this mushroom contain iodine. So I must say that it's better to avoid this kind of mushrooms that has certain heavy metals or chemicals.
Thank you very much,
Dear
sir.
sir, we are short of time. Take further questions at the end of the session. Please forgive for this, because we have few more talks coming in line. Now, let us close this session. We genuinely thank Dr. Sam for explaining every answer in detail with much more clarity. And we are delighted to have many professors and everybody listening to you. And now the technical session will be conducted by Dr. Gunjan. Over to you, Gunjan, for the next talk of Dr. Damodara Shenoy.
Thank you, Raj.
Thank you
And
very
hello,
much.
everyone. It's my pleasure to welcome our next speaker, who is also my PhD supervisor and mentor. So today he will be talking about species and species complex. And I would like to briefly introduce him. Dr. Belle Damodara Shenoy works as a principal scientist at CSIR National Institute of Oceanography Regional Center, Visakhapatnam, located in Andhra Pradesh, India. He did his PhD in mycology at the University of Hong Kong. After a brief postdoctoral training in Canada, Dr. Shenoy joined CSIR India at Institute of Microbial Technology in Chandigarh. In 2013, Dr. Shenoy moved to CSIR National Institute of Oceanography, India. And he's a passionate mycologist, promoter of mycology, and a co-founder of MycoAsia. Welcome, Dr. Shenoy.
Thank you very much, Dr. Gunjan Sharma, for your kind introduction of me. And it has been a kind of a very intense, almost 2 hours, 1.5 hours now. I promise it will not be less intense, because what topic I'm going to talk about, I'm going to talk about species versus species complex. It's kind of a very basic type of research query, and it will definitely be in your benefit, if you listen to it very carefully. Thank you very much. And first of all, what is a fungal species? Fungal species. What is a fungal species? Sounds like a very simple question, but the answer is not very, very simple yet, especially the advent of DNA sequence technologies and other stuff. As you know, species is the basic unit of biodiversity. So what is a species? Jokingly saying the species is what our taxonomy says it is, because taxonomists are the authorities when it comes to describing new species or describing new taxon. So, but when a taxonomy says something, he should have some credible evidence to support his hypothesis, because in any taxonomic category is a hypothesis. For example, species is a hypothesis. We test it and we prove or disprove it. Same with the genus, family, order, class, phylum, kingdom, domain, for that matter, life. So conceptually, species are segments of evolutionary lineages that have evolved independently from each other. So basically, the independence from the closest taxa or closest lineages are very important. Independence in terms of genetic barrier, independence in terms of other factors also. How do you test the independence of a set of organisms? There are some tools. We use molecular tools nowadays. So conceptually, as I told you, the species are segments of evolutionary lineages that have evolved independently from another. But when it comes to recognizing whether some individuals should be considered the members of the same species or different species, how do you do it? There have been 4 different kinds of species recognition criteria. First one being morphological, where you look at the external appearances of fungi like hyphae, spore, ascomata, and all. It has been in fashion since till 1990, but because of DNA sequence data and other developments, phylogenetic species recognition has taken over the morphological species recognition criteria. Biological species recognitions are not that easy to apply in fungi, because many of the fungi may not reproduce in artificial culture media. Ecological species recognition also has limited, limited application when it comes to species recognition in fungi. So morphological plus phylogenetic and combination of both have been in a great fashion recently, especially since 1990s. When you use one gene for species recognition, that becomes quite limited, in the sense limiting kind of. One gene will tell the history of that gene itself. But to exaggerate kind of extrapolate it into whole speciation event in that group will not be that reasonable. So therefore we need to use more than one gene region. So we then go for multi-locus sequence analysis. When we use these genes, multiple genes, we make sure they share similar evolutionary history. That means genealogical concordance should be verified and should be tested. All right. So, and these genes should be physically unlinked on the genome. So when you use multiple unlinked genes which show phylogenetic concordance, so this can be used to prove the lack of genetic exchange within a group of individuals. So thus we can prove the evolutionary independent of lineages. When we prove the evolutionary independent of lineages, we define the species boundary. So defining a species boundary is a very complicated matter. You can identify a species based on morphology, or you can identify based on morphology plus gene sequence data. But defining species boundary is altogether different ball game. I'm going to tell that in a while. Please wait. Before I have already told conceptually what a species is. Now, what is a species complex? So especially when you have specimens, especially fungal specimens that exhibit identical morphologies or very close phenotypic traits, yet they show very distinct genetic profiles, especially in the multi-locus sequence analysis. They show distinct profiles. How do you treat them? How do you deal with them? Morphological, they are reduced. But when it comes to phylogeneity, they have got very distinct, distinct profiles. How do you deal with such things actually? We work on Coltotrichum gliosporidis sensulato. In simple words, Coltotrichum gliosporidis species complex. There are other species complexes, for example, Aspergillus niger species complex, Physaриum subglutinans species complex. Okay, so when we can use genealogical concordance phylogenetic species recognition criteria to separate phylogenetically distinct cryptic species, which are morphologically very primitive. So that has been the strength of genealogical concordance phylogenetic species recognition, in which we use multiple unlinked genes and which are phylogenetically which have phylogenetic concordance. All right. Talking about Coltotrichum gliosporidis, it is a very common name. It was associated with more than 450 plant genera. That means anything you found on many of the plant genera, which looks like this, it would be named Coltotrichum gliosporidis. The species was loosely defined till 2008. The DNA sequence from the type material was not very much sure. So Paul Cannon and the team actually designated a lectotype. Because it's a very complicated matter. You just read the paper, you will understand how complicated the history of this particular species. Because this species was described in 1882, and there was no specific type material described. Okay, so they Cannon et al. Actually they designated a lectotype. It's a kind of designating one material as the authentic material or reference material for taxonomic purpose. They also designated one eptype. Eptype is an extension of the type material which can give additional information like DNA sequence data. So finally, we have DNA sequences, what you call credible DNA sequences from the type material of Coltotrichum gliosporidis. So what you do, when you use the type sequences of gliosporidis, the story changed actually. So especially in the tropical countries, what we thought gliosporidis was not at all a gliosporidis in a new sense, in a new definition actually. So many of the fungi which were identified as Coltotrichum gliosporidis, causing diseases in tropical fruits, were found to be not Coltotrichum gliosporidis, something else. For example, Coltotrichum siamens, Coltotrichum fructicola, Coltotrichum asianum, Coltotrichum hori, Coltotrichum kahave. So, the siamensis. So you have more than one name for the morphologically similar fungi. All right, but they are genetically distinct or phylogenetically distinct. So it has led to kind of interesting research. For example, Belle and team actually they did a fantastic work on phylogenetic redefinition of Coltotrichum gliosporidis. So they used multi-locus sequencing phylogenetic analysis. So here is the Coltotrichum gliosporidis in a very strict sense, in a newer sense, and here are the phylogenetically distinct species under Coltotrichum gliosporidis species complex. So previously one name, now at least 23 names. All right, very interesting. All right. So this is kind of resolved that Coltotrichum gliosporidis is a species complex has at least 23 phylogenetic species. All right, very interesting. This has implications in plant pathology, plant breeding. All right, for example, you used to identify this fungus as gliosporidis, this also as a gliosporidis. So I was talking about, people misidentifying Coltotrichum fructicola as gliosporidis before the designation of, authentic DNA sequence data from the type material. So very interesting stuff we got after this reorganization of the taxonomy of Coltotrichum gliosporidis. Very nice. Gunjan worked on Coltotrichum siamens from Indian materials, Indian plant substrates actually. as those are mostly endophytes and pathogens. And she used a different gene, what you call APMAT marker. All right. APMAT marker actually showed lots of genetic diversity within Coltotrichum cyan species complex. So one gene could actually provide lots of information about genetic diversity under Coltotrichum cyan species complex, what we thought. All right. We did multigene phylogenetics. We also described new species under what we thought Coltotrichum species complex, cyan species complex. But Professor Chai Lai and his team from Beijing, they did a fantastic work. They found out that the single gene phylogeny, what we showed using APMAT, and the 8 gene phylogenies that they produced are almost congruent. I mean, they were similar, but when they used what we call, coalescent methods, these are the methods by which you can actually define the species boundaries. You can tell whether the recombination is happening or not. There is a genetic barrier. All these things can be, kind of revealed using coalescent methods. It's a kind of phylogenetic analysis, sophisticated phylogenetic analysis using multi-gene sequence data. What they found was that the Coltotrichum siamens sensulato is not 7 species. That there were 7 species under Coltotrichum siamens species complex, but Chai's team demonstrated that Coltotrichum siamens is a single species. There is a genetic barrier among the 99 isolates or 90 isolates he used. So whatever the 7 species described under Coltotrichum siamens species complex found to be synonymous. So all the 7 species were merged, and now we have only Coltotrichum species, Coltotrichum siamens. Simple, even though you use 8 genes or 1 gene, which may be congruent. Finally, you have to use what you call coalescent methods, by which you prove that there is a genetic barrier and recombination is happening, or you define the species boundary in a very sophisticated phylogenetic analysis. So lesson learned was that in addition to the genetic studies, they also did the mating test. So they found that members of the Coltotrichum siamens, in sensulato, they could produce the sexual structures on pine needles. All right, in the laboratory condition, kind of. Okay, so that proved that these fungi under Coltotrichum siamens sensulato could actually interbreed and produce the the sexual structure. That means they are a part of same species, not 7 species as we incorrectly pursued when we did not use coalescent methods. So we learned our lessons. I would like to share with you our lessons. When you deal with fungal cryptic species, please diligently document the morphology. Okay, even though morphology may be primitive or kind of not well developed, but look for new morphotaxonomic characters, because it might actually save you from lots of trouble. You don't have to use 8 genes, 6 genes. If you have some nice new morphotaxonomic character which can differentiate this species, that would be wonderful. It will be less expensive to do in the practical life, actually. When you do phylogenetic analysis, simple rule: include diverse numbers, include type sequences, include non-type sequences. When you are defining a species boundary, it is a good practice to include members from all over the world, if possible. Chai's team used fungal cultures from many countries of the world, included type cultures, non-type strains. So, as I told you, we need to use multiple unlinked phylogenetically rich genes. Phylogenetically rich genes. One observation we made from Chai's work, that even though he used 6 genes, they use 6 genes to reconstruct the phylogeny and redefine the species boundary. the phylogenetically informative characters were only 150. In the now the era of whole genome sequence data, what if you add, instead of 150 phylogenetically informative characters, we make it 1,500, phylogenetically informative characters? Would the species boundary will be the same, or whether the species boundary will have to be redefined again? So there is a kind of a mental battle, whether it is a species or a species complex. It all depends upon the evidence you produce to the world. Today they have about 150 phylogenetically informative characters. Tomorrow some same people or other set of people come up with the evidence of 1,500 phylogenetically informative characters, and redo the analysis. Probably the story may be the same or may not be the same. So better to use multiple unlinked phylogenetically rich genes. Of course, we made a mistake in Gunjan's paper. We did not check for the phylogenetic concordance of the genes, especially we combined APMAT with other genes. I think that was a mistake. But nevertheless, that Gunjan's APMAT tree had lots of genetic diversity, which was similar to the 8-gene tree reported by Chai's group. Of using coalescent method, you can prove the lack of genetic exchange. That means these are as one species, or then finally you verify evolutionary independence of lineages. So other than genetic data, we can do culture. It is a good idea to do mating tests using cultures or plant materials in case of plant pathogen. So with this, I conclude my talk. I thank the organizers. I thank my organization, CSR NIO. Most of the work on this was done when I was in CSR intake, and Gunjan was my student there. And I also would like to acknowledge the contributions of my other students and collaborators. They have kept me going with this kind of research. These are my contact details. You are welcome to contact me anytime. Thank you very much. Over to you, Dr. Gunjan Sharma.
Thank you, Dr. Shenoy, for a wonderful and insightful lecture and helping us understand about species and species complex. So I would like to invite the audience for any questions, please, if they have.
Sir, I have a question to ask you.
Yeah.
Am I audible?
Yes, you are.
Okay. My question is, how does the habitat help in identifying different species of the same species complex? Like, do they have the same habitat or they have different habitats? I want to know that.
I mean, regarding Coltotrechum gliosporidis, they actually, Coltotrechum gliosporidis sensulato, they infected the diverse plants genus actually. Okay. When it came to Coltotrechum siamens, we didn't find host specificity. They could infect diverse plant species. So I don't think host had any role in speciation there. I mean, finally it's one species according to the recent data.
Okay, so thank you.
Thank you, Pratika.
So, Komera Surya, your question, please. Jackson, you have a question? You raised hand.
Yes, sir. Thank you, sir, for the wonderful presentation. Till now, I'm not very clear about the species and species complex. And I don't know if my work is related here, but my work was on wood decay fungi. And while I was doing my work, I came to, I stumbled upon certain species which I think or assure that it can be critic species. So how can I identify them, sir? Like, how can I identify them as critic species? Because morphological wise, I have checked everything. Everything is very similar, 100% similar. Only in the culture, on the hyphae, I found some differences. But other than that, it is like completely same. So I have sent it for sequencing. So is it the only way to identify it now?
Thank you, Jackson, for that wonderful question. It's not easy, if it is a cryptic species. You will have to accept that it will not be easy. First of all, you start with ITS, okay? Then for the identification, definitely it will be misidentified. Okay. Then you go for multi-locus sequencing. So then you will get the phylogenetic position of the tree, position of your fungi. Okay. If you find genetic diversity, probably these are pretty different when it comes to genetics, but morphologically very similar. Okay. So you cannot use morphology. So to separate them, you have to use, as I said, multi-gene sequence data. All right. Then you have positioned them on a phylogenetic tree. Whether they have stopped exchanging genetic or genetic material or this closest lineages, you have to prove that using coalescent methods. We did not do. For that matter, many mycologists did not have a practice of doing coalescent analysis when we were working on culture tracking. So this is a kind of a very sophisticated analysis. Day to day, you don't do this kind of analysis. You rely on mostly one gene to identify, hoping that it is a correct identification. I hope I did not demotivate you.
So,
Any questions?
If you allow me to follow up, so sir, do I have to use only the ITS, or can I use RBP2 and TEF also for identification?
Jackson, it depends upon your group actually. What is the state of the art in that particular fungal group? What other researchers have been using? So you have to build upon the existing data. Okay, if they use 6 genes, you start with that. All right. But if you find these 6 genes are not really phylogenetically rich, probably you can look for newer genes and redefine the whole species boundary. It's going to be very exciting, but it will be a little bit challenging also.
Thank you so much, sir.
Thank you, Jackson.
Hello, Dr. Shenoy.
I have a question. Can I come in, please?
Hello.
Can I come in, please? I have a question. This is Dr. Dhanokar from Symbiosis.
Yes, please.
Yeah, sir, in today's advanced techniques of fungal identification or nomenclature, like DNA sequencing or phylogenetic studies, still does any morphological identification still holds any role in today's fungal identification?
Oh, it is important to document what you see.
Yes.
Very important. You cannot ignore that. You start with morphology. If the morphology doesn't have enough morphotaxonomic characters,
right,
then you just try then you rely on the phylogenetic characters. Then you try to combine the both phylogenetic and morphological characters. So you can map the characters on the morphotaxonomic characters on a phylogenetic tree and try to make sense. Morphology is essential. I am not saying you should skip it. I mean, it should be the first thing you should look at in a fungus.
Yeah, so accordingly, whatever genera-wise or species-wise identification keys, morphological keys where they were given, are they still valid with that, and how do they still differ with the current DNA sequencing, or they match with the current DNA sequencing parameters?
It's a very
many species were identified based on the morphological keys, and many mycologists still do that.
Oh, it's very fair enough. We don't have type materials for so many fungi, and
yes,
without having type materials for so many fungi, there wouldn't be type sequences. Even if you try to do molecular identification, where are the type sequences? Yes. Or it's still playing in the dark sometimes. All right.
Thank you.
Thank you.
Pushpavati.
Good afternoon, sir.
Hi, Pushpavati. Yeah.
Sir, when we identifying this cryptic species using multi-locus data, usually any species with less divergence in that locus. If the locus has less divergence, if we construct the phylogenetic tree, it will fall into the same class even if it was a different species. So, can you hear me? Hello?
Yeah, I mean, my point is very simple. Takeaway message is very simple. Can you hear me?
Yeah, sir.
My takeaway message is very simple. When you start working on species complexes, you just start with the available data from state of the art, what has happened so far. Okay. If the already used or the genes which are already in use, if they have phylogenetically rich regions, that means they have got very good phylogenetic signals. Definitely, it will differentiate into multiple species. Okay. Problem comes if the genes used are not phylogenetically that informative or that rich. Then you may have to look for new genes from the whole genome sequence data or some other literature. Of course.
Okay, sir.
I think it comes with the experience, Pushpa. Yeah.
Okay, sir. Sir, knowing the genetic distance by calculating by using already present existing data can help us to differentiate or to decide whether the particular locus is good or not, to discriminate our present species.
Yeah. When you deal with cryptic species, you just read what has happened so far since 1990, at least since 1990. So it will give an idea whether to use those genes or you should look for new other genes. All right. So literature survey will make your life easy. Yeah, a good literature survey will make your life easy.
Yeah. Okay, sir. Thank you, sir.
Thank you.
Dr. Gunjan, can I ask a quick general question?
Yes, please.
Yes, Dr. Shenoy, thank you for your analysis of species versus species complexity. I enjoyed it. Now, my general question is, see, you came up with something which has 23 different species identified after a great analysis of including phylogeny, morphology, and other characteristics. Now, do you have, and you made an important point in your presentation that time, saying that the differentiation of this is very important because these are endophytic parasites, I understand, that any treatment method should be based on some of these understanding of the species. So do you have a fungicidal biogram for all these, one of these complexes that you mentioned, given the fact that the coalescent method has reduced a couple of or some species from 23 to less? What's your take on that?
Thank you, Dr. Ramaya. Thank you very much for joining in and asking me this wonderful question. See, we had the transition from morphology to sequence data in the 1990s. Okay, people have been trying to incorporate other data, what we call secretome or phenome, for example. All kind of external, all kind of non-genetic characters. We don't have kind of what you call fungicidal, profile for these important plant pathogens under Culturetrachum gliosporioides, for that matter, gliosporioides species complex, for that matter, the lineages under Culturetrachum siamens, Culturetrachum siamens as defined by Chai. So it would be interesting if somebody did and try to map the fungicidal profile on the phylogenetic tree, especially after the coalescent method of analysis, but it is a long way to go before we can say it will be useful or not.
No, I mean, solving this great problem is very welcome, but that should be kind of used in the fields. That's my thing. It's a great point what you made, and that's what I appreciate. Thank you. Wonderful.
Thank you. I mean, in the field, one example I would like to give, without the fungicidal profile, the reorganization of Culturetrachum taxonomy has been very beneficial. In Thailand, they used to use one Culturetrachum species for plant breeding. Okay, I don't know how successful was their plant breeding program to find the new varieties or plant disease resistant. But after the phylogenetic reconstruction or redefinition of the species complex, it was found that they were using a wrong phylogenetic species in plant breeding. It's kind of not fun to use wrong fungus for plant breeding. So that is a field level application of a very good credible taxonomy. Thank you.
Any more questions?
Hello.
Yeah.
Hello, Dr. Shri.
Hello, sir. Hello, sir.
Good afternoon. Good morning, even. And congratulations for your nice lecture at outset. And I have just a little query, you can say, or just I want to know something. Because these Coleotrichum are isolated from plants also as an endophytes, and sometimes question come that have you checked whether this Coleotrichum is pathogenic or not? Though they are growing in healthy plants, so we assume that they are not pathogenic. But do you know any methods through which it could be decided whether the isolated one is pathogenic or non-pathogenic in vitro? Because Koch postulate will take time. So is there any method through which it could be decided whether it is pathogenic or non-pathogenic?
Oh, that's a wonderful question, Professor Karwat. Thank you very much for that. I mean, it would be wonderful if you could have some method without doing cost postulates to tell whether particular fungal isolate is a pathogen or where it has the tendency toward pathogenicity.
Yeah.
It would be very nice, but maybe transcriptomics data, maybe proteomics data can help in this regard. But it's the research of future, I believe. Thank you.
Okay. Okay. Thank you, Dr. Shenoy.
Thank you. Yeah.
So thank you, everyone. So if there are not any more questions, let's proceed to our next speaker. He's Dr. Rajesh Kumar. He's working as a scientist at National Fungal Culture Collection of India, NFCCI, at Agarkar Research Institute, Pune. He did his PhD from Forest Research Institute, Dehradun, on biodiversity of plant pathogenic fungi of the Western Ghats. He also did his postdoc with CABI, UK, which was previously known as IMI, and he explored Indian Himalayas for the fungal biocontrol agents of the Himalayan balsam and hedychorium species. And he's regularly publishing articles on fungal taxonomy in very good journals, and I'm looking forward to his great talk. So today he'll be talking about role of fungal culture collection and fungaria in mycological research. Over to you, Dr. Raj.
Thank you, Gunjan, for the warm introduction. And I thank Venkateshwara University and MycoAsia for this opportunity. It's rather a dry topic to start with after such a scintillating lecture from Professor Dam. And let me take you through the role of culture collections and herbaria in mycological research. One of my mentors always used to say, no fungi, no life, no future. She's Ami Rosman from UST. As a part of the largest culture collection, fungal culture collection of India, the National Fungal Culture Collection, which is situated at Agarkar Research Institute, Pune. We are currently having around 5,000 accessions, and we are focusing on the repositories, services, R&D, and HRD, which are the key components of a culture collection. So coming to the biological diversity, you know that the Rio conference has set up the targets for each countries to conserve their biodiversity, components of biodiversity, and their equitable sharing of the benefits arising from these biological resources. When we talk about the CBD and the countries ratified across the globe, around 160 countries are ratified in the beginning, and they must also insist about the in situ conservation method, that is on-site conservations as well as the ex situ conservation. When we talk about the culture collection, it's coming under the rules of ex situ conservation protocols of the CBD. Then what exactly happened was, initially the conservation priorities were given by United Nations. You can see the symbols where plants and animals and insects were got some kind of emphasizes, rather than the fungi and bacteria or the microbes were kept together under the heading like microbes. This was affecting the international arena for research in fungal diversity. So rightly, okay, are the offense of the Rio. It is because fungi as the second largest organism spreads with about 99 phyla at that point of time. Currently, they are in 19 phyla, and they were not recognized for the biodiversity conservations. After this, they were alarmed from the British mycologists to take up the conservation initiative through various forums, and International Society for Fungal Conservations established. And now today we have international societies like Species Survival Commissions under United Nations, and we have even the Global Fungal Red List initiatives. Even you can contribute a fungus from your area following the protocols given by the Global Fungal Red List with the guidance from Species Survival Commissions group. So why we are talking much more about the fungal diversity and their application and the culture collections and fungaria, it's because there are amazing potentials of fungi. You can refer to Kevin D. Hyde's paper on the amazing potential of fungi, 50 ways we can exploit the fungi industry. It has almost every topic on the fungal applications has been covered in this, including the agriculture, medicine, industry, and even climate change discussions were made. You can avail this paper from Tillini or Professor Sam. He's sitting in the panel. And also the magnitude of diversity. You know that when the first proposal came around 1991, professor told that Professor Hawksworth proposed that there were around 1.5 million fungi. Later on, he revisited this in 2001, and further, the revisit in recent 2017 happened about 2.2 to 3.8 million species. What we know currently is only 1.5 lakh species across the globe, and among that, we have only less than 10% of the biological resources. That means cultures are available in the culture collections, and we are also lacking qualified mycologists for identification of all the species across the groups of fungi. We are lucky to have Professor David Hawksworth at NFCCA, and we had extensive discussions on the nomenclature as well as the fungal taxonomy. He will be, the next session of IMC, will be proposing even DNA as the type material, which was a controversy and got rejected in the last convention. So what is the culture collections? What exactly the culture collections are doing for strengthening taxonomy? We, as a part of culture collections, we decisive roles in ex situ conservation of our nation. So we have 5,000 cultures. We supply authentic strains. We are bound to identify the fungi accurately and help the researchers across our country and preservation of microbial taxa and strain for future. Long-term preservation protocols are needed because we are losing the land management as well as we are losing the biodiversity due to land management and climate change, and also the modern tools and applications. Molecular tools are in state, so there are plenty of research going on and changes are happening, which is already discussed in the previous talk, as well as there are a lot of changes are coming up in taxonomy of fungi in a global arena. When we talk about the biological resource centers or culture collections, we must understand the Budapest Treaty under World Intellectual Property Organization. Actually, a culture collection can be of different kinds. That means the International Depository Authority under the Budapest Treaty recognized culture collections will have 3 sections under patent deposits for IPRs, safe deposits for persons who are wanted to save their confidentiality of the cultures for long term for specific research purposes, as well as the general deposit, which we normally do in the general deposit. The culture collection may publish your strains for the public and may supply it through an MTA. We are lucky to have a few wonderful culture collections in our country, like MTCC, Microbial Type Culture Collections, MCC Pune, which is now known as NCMR, as well as the Agriculture Department of IAR sections taking care of NAI MCC. So what is the exact pattern of culture collection, how they are governed? It is actually the International Union for the Microbiological Societies set up the rules and guidelines for the WFCC. They have their own guidance, as well as, WFCC maintain a database and data sets for all the culture collection across the rules and regulations, culture collections across the globe, as well as give designations and numbers for each culture collections. Also, when we talk about the culture collection, the data maintenance actually happened under the WFCC Mission World Data Center for Microorganism, which is currently situated at CASBJ. They are actually maintaining the culture collections data for and even a global catalog for the microorganism. That means all fungi, bacteria, cell lines, actinomycetes, and all. And NFCC is one of the only recognized World Data Center culture collection, which is designated as WDCM 932, which is situated at Agarkar Research Institute. I'll just give a brief about the World Federation of Cultural Collections and what they're exactly doing in their database. When you visit there, you can see CC info means the culture collection informations you can get. There is a global catalog microorganism. You can search through different microbes available across the country for the application studies and what is their current status and all details through that, and analyzing the biological resources for the citation. That means these culture collections, how many publications came out of it, patents and related informations you can avail easily from the culture collection. When you are aware about WFCC and WDCM, you can even search for your colleagues who are doing a particular research in your area of on a particular organism, and you can put proposal for collaborations as well. There are also reference strain catalog available with WDCM, and these rules for culture collections are actually set up by OECD guidelines, and they follow WFCC guidelines also for setting up the culture collections. If you are interested to propose, the fact of the matter is we have around 27 culture collections in India registered under WDCM, but unfortunately, very few culture collections are following all the standard protocols, as well as maintaining the cultures in a standard protocols, which is recommended. And also, there are few criteria, for example, the layout of the labs. For example, you may be aware that COVID made us to aware about the BSL-3 labs, and you send samples to Pune. It is because we have 5 BSL-3 labs available here. There are criteria for setting up the building and structures, instruments available, and the core and buffer zones for the culture collections. We must have at least basic all these criteria to handle the normal pathogens, level 1, level 2, level 3, and level 4. So, coming back to the culture collections of global catalog, I just wanted to give a brief for your understanding. You see, these many culture collections are already members of WDCM, and you can find the codings for each of them and the countries mentioned here around. From India also, we have NFCCA as a part of this. And when you search in a global catalog microorganism, for example, any name whichever you wanted to see, the cultures available across the countries. For example, Bacillus, it's given. For example, if you're giving Aspergillus, you can find that these many options are available to understand where these cultures are present. And also each culture collection wise, you can search it, each organism wise, you can search, each continent wise, you can search, and also you can filter. There are a lot of options available to understand the diversity of cultures present in different culture collections, which are recognized by WFCC and WDCM. Even continent, geography-wise selections you can make, even particular strains. From my culture collection, I was searching for Aspergillus flavus, and we found that these culture collections are available. Cultures are available with us, where from these details, where from these are isolated, what's the host and other details. And also, we can have an MTA to get these culture collections from the curator of National Fungal Culture Collections. So there are pure cultures which is valid and recognized across the globe are available in your culture collections. So that make the research for the coming up generations quite easy to work on deeper genomic studies and application studies, because these are authenticated cultures. Also, there are patent details available for every microorganism, whichever considered for the patent studies. And you can see these numbers, read about it. If you are selecting some fungus or for your research, future research, you will understand that there are already few patents taken over these. these details can also access through WDCM details. For example, you can see a fungus details with the patent details, and all the details are given here. Similarly, culture collections are also observing over the performance of each cultures and the diversity of cultures and countries. Even they are even determining how many sequences coming out of these collections through WFCC publication reports and the genomes developed. India is one of the leading country now developing maximum genomes in NCBI and the world platforms. Along with China is the leading country along with the USA ahead of India. So here you can see NFCCA. We have around 1,300 strains kept in the global catalog of microorganisms, and details are given. And as we have some regulations applicable in India, National Biodiversity Authority, we might have restrictions for using this culture for application to the third parties outside the country. Rather, these cultures are available for freely for Indian citizens currently for applications, and there are provisions under National Biodiversity Authority. If you are checking their website, you can find it for sending it for taxonomic purpose. So there is a possible collaborations possible across the continent for only for taxonomic purpose currently. So when you consider a deposition of culture or working on any particular culture, you want to deposit, you must have the following details for each data for each culture, which helps to understand the strains, nature of the strains, and details of the strains, its ecology, its origin, geographic origin, which are the favorable temperature you prefer that, and what are the applications you are going to study. When you share with these kind of information help the next generation, because they will be focusing on particular research based on your biological resources, which may lead to the greater study in future. Then what are the methods we generally adopt for preservation of fungi? It is you can read about this book of Smith and Onions. They give short-term, long-term, medium-term searches, preservation methods. I am not going into the details of this because it's the standard protocols for every culture collections to follow. If you want such details, you can contact me personally. I will share with you all the details which we were following at National Fungal Culture Collections of India. So we must also have some infrastructures for capturing the culture collection storage. So walking chambers are essential. We must have lyophilization units for fish dyeing, and all these do's and don'ts I have mentioned here. We must have cryopreservation cans, as well as the supply of liquid nitrogen in your facility. So it's a costly affair when it comes to when preserving cultures for long term. So, but there are possibilities that we can conserve our valuable biological resources, fungal biological resources of India for future generation in a long term, like 20-30 years. If you want to you want to keep your cultures in a very good conditions, this protocol, cryopreservation, liquid nitrogen storage, is one of the best method. So what are the other services which we provide at National Fungal Culture Collections? We have 19 services targeting supply of cultures, supply of industrially important cultures, accession of fungal materials, and we do a little bit about the barcode gene sequencing and secondary barcode gene sequencing. We do support researchers for many other aspects also. Some students, when they come up with problems for morphology, brief description, digital microscopy, we have some packages for them, and also we are supporting the Indian students, academicians, and researchers for achieving their quality results with standard authentication protocols following at ARI. And okay, this is the first part of my presentation, which was focusing on the culture collections and its application, how a culture collection can help the researchers across India and globe, as well as I have to talk about the fungarium, which is an old subject, but which is much more essential. There were questions about the taxonomy of fungi. So when we talk about the taxonomy of fungi, we must aware that there are 300 years of continuous effort of morphological validation. This morphological validations, you must understand, this is based on the whole genome of that fungus and its expressions to the particular ecological parameters. That is why they have evolved in such a way. So The herbarias have historically served as a repository for fungal diversity. When you talk about the herbaria of India or globe, you must understand that represent the hundreds of years of fungal heritage of the nation. So when they have pertained to some diversity of fungi, you should understand that this will give a lot of information for the future studies, as well as they understand they will work as a reference stand for your any kind of taxonomic clarification studies, the base material for taking up a few morphological characteristics, as well as taking up the reference for proposing new species. All these reference materials across the countries are available. They are listed also. But you should understand, prior to the 1955, there were no mandate for deposition of type materials. So prior to 1955, you may not able to get good fungal herbaria or fungaria across the globe because there were no mandate. But with the botanical code established in 1955, there is a current scenario that we have to have a deposition. There is a mandate now as per ICN rules and regulations. So you must understand, when you talk about the material, somebody was referring, there were only one material. Actually, if you want to serve, if you want to propose a species, and the world should recognize it, and at least few mycologists, if they want to get it, you must make an exicata. Exicata means there must be a holotype and its isotypes, and the copies of this as like topotypes or similar materials, and you must be able to deposit in multiple places. Otherwise, you must try to achieve the multiple collections from your place or host species present across the habitat, so that you can present your material. There is no restrictions for publication of a species based on single material, but it would have been better. It is better that there are some directions from ICN that if you are depositing a material based on one singular material, you must mention that in your publication which you are making. When we talk about India, we have HC Hyo, Herbarium Cryptogame India Orientalis, as well as Ajuraker Mycological Herbarium, which are the pioneers in the preservation or for the materials of fungaria. So we have, you can see here, 49,000 of disease specimens. When you are taking up plant pathology as a topic of your research, there is a place to go and see and what kind of diversity existed in India. We have a past to define. Similarly, when you come to the Ajuraker Mycological Herbaria, you can see 30,000 lichen specimens from majorly from the Western Ghats are conserved here. You exactly can define what kind of diversity existed across the Western Ghats among the different states. What are the host plants associated, and what you can also get some few books related to this. So these information, with this background, if you start your research, you are talking about diversity of the fungi as well as application. You will be, added up with a lot of information. The Herbaria Cryptogame India Orientalis, they have even come up with the publication of their catalog, which are freely accessible. So this way, Herbaria helps the researchers for understand the historic diversity present in those particular biodiversity hotspots. We have two biodiversity hotspots you should understand. For example, for lichen and BRI Lucknow and AMH India, like AMH Pune. If you go there and visit their collections, you will understand how intensely researchers are contributed to the lichen diversity of India. So these are some herbaria. When we talk about the herbaria or fungaria, you should know which are the codings, because in publication you may not able to see the whole number of these things, but rather than we use codings. So you can see that the Berlin collections is mentioned as B and Munich collections is mentioned as M. Likewise, AMH is Ajuraker Mycological Herbaria, in HCIO for our Delhi ITCCIR collections. When we talk about the materials, when you define a species, new genus, it's mandate of to define a holotype, which is a single specimen representing the name-bearing type, where from the name is originated. And if you want to know the rules and regulations, go to ICN and go through the chapter 2 of status verification, priority name, section 2, and typifications, and the article 8. And the article 8.4 says that type specimen, name of taxa must be preserved permanently, may not be a living organism of a culture. So it must be a dirt material rather. So it can be, the addition which happened in the Melbourne Code in 2011, 2012 was you can also use a culture in a metabolically inactive state. That means that lyophilization and even liquid nitrogen cultures can be considered with the advice from the expert, as well as you have to mention these details exactly in your publication, that these are in metabolically inactive forms and those cultures are deposited in a particular culture collection with reputed designation. So when we talk about how you link the type material and or material or fungarium with culture collections, this is the example that when you deposit a holotype, if you can culture that particular strains, those culture are the living representative of the holotype or the name-bearing type or the fungus, those are should be submitted. Those strains should be submitted in recognized culture collections, either in an IDA or a recognized World Data Center of Microorganisms, separated culture collections. For example, here you can see we have published fungus recently in penicillium section, and we have all the details of the cultures in 10 different media, 3 different temperature, and we defined it through a combinations of genes for identification as well as phylogeny, and we did concatenate analysis and further published this in a reputed journal. So here you can see that while defining the fungus also, there are some basic rules which you have to follow. You have to name and you have to give author citations, classifications, all media details, and details of colony characteristics. We say macromorphology, then coming to the microscopy details. Then you can see here type details are mentioned. All these type details who have collected it, what is the holotype, what is the X-type culture, what are the gen bank numbers, what are the additional materials reviewed for establishing these researches. So you must have details of all these parameters to emphasize that your collection is a valid, legitimate, published species. Another example I wish to show you about the Saccardo's collections. He has collected the Pesiza around 1874-75. Those materials were present in Italy, and few researchers attempted to get DNA through a highly, tools. A tool like isolation kits were developed, and internationally it is available now from this much old samples because of the preservation quality. When we talk about mushrooms or specifically lichens, the preservation of quality, there are protocols for doing. Experts can only understand at which temperature it should be dried and focused, so that even DNA didn't get damaged. They could do it with a little bit next generation sequencing platforms like Illumina and assemble the data, and they come up with around 23 ITS sequences for this out of the 36 specimens collected during 1875. And you must be wondering, who is Saccardo? Saccardo was the person who is establishing at least a Silog fungorum and naming these fungi based on the spores type, color, shape, and septations. He was actually the first person attempting the earliest stage of the early stages of fungal taxonomy, how a fungus can be defined based on the spore morph. Even currently, when we talk about the Hyphomycetes group, even Seyffert's book, you can see, Carmichael's book, you can see that the fungi are arranged based on spore morphology: Amero, Tetimo, Phragmo, Dictyos, Colico, Helico, Kairos, Tauro. This spore morphology, if you need also, you can see the development happens. Even our legendary Professor C.V. Subramanian, he has come out with the thought that the secession and conidial like genesis type should be included in the taxonomy part. When you study in details about the Hyphomycetes, Coelomycetes, you can understand that what kind of conidiogenesis happens, what kind of conidiophore developments, what kind of conidial types are based on these circadian principles. Even you can see a recent publication from Martina said that circadian has described these fungus as stachybotrys, and stachybotrys were polyphyletic, hopefully. So memnonial stachybotrys complex were there. So they delineated it as a new species, because when paraphily appears, somebody asked a question about when distance were minimal. Some cases, the same similar looking fungus can become a different species also. They are genus also, because they have such derived genomic data supporting, and they have been compared. These kind of studies are possible when there are lot of data and details available in that particular group or family. Similarly, we had an experience with Aspergillus pongamiae. I'll just skip through it because I know that I've taken much more time rather than allotted to me. We have proposed a new genus combinations for Aspergillus pongamiae. Everybody in India knows that is a pongam fungus is Aspergillus pongamiae. We have studied the morphology, we have gone to the sections, measurements, and each and every character in detail for 3 years. Because when we first proposed the sequences and these differences to the international panel, they said that you might have missed the characteristic of genes and sequence may be wrong. So we did collections in 2017, 2018, and 2019, and even a collection in 20, and validated that all these 3 years we got quality sequences from these materials. And we proposed then we understood the clumsiness around the morphological identification, why it was wrongly identified, because it was initially identified as Fusicladium. When we search about the Fusicladium and its positioning, it was not a part of Mycospheruliaceae family. Rather than it is family of, currently Fusicladium is categorized under Venturiaceae. So then we went to the other synonyms, and then Mycospheruliaceae synonyms are there. Members, synonyms like Passalora, Aspergillus pongamiae, Aspergillus pongamiae pinnata. Even Carver sir was sitting here, he has identified it. The confusion happened because the plant name nomenclature changed. It was Pongamia glabra earlier, later become Pongamia pinnata. Such confusions will added up to the naming of fungi based on the morphology. So when we look at the exact story of this, these are Indian and Sri Lankan species, you can say. I cannot say exact origin, but rather than the collection-based study we did, then we understood that Seidorf and Seidorf, their German scientists, came to India during 1930. So these names were 100 years old, and we were resurrected these names that rather than put it as a new combination, because after studying all the type materials, we had an access to the syn type material. That means 1 or 2 biological types, which when they lost the holotype, we designate syn types. And similarly, the multiple collections which is made from the same locality were present. This has been done with the support of Uwe Braun, one of the legendary mycologists from Germany, because those collections were available at Germany, in Berlin, Munich, as well as in IMI. Few samples were available. So those data were aggregated by Professor Uwe, and these collaborations made through Professor Krauss. He was the ultimate person for Mycospheruliaceae. So here you can see, after 120 genera, if you're adding a new genus to the complex taxa, this Mycospheruliaceae were known as dustbin group, because everybody used to dump species into these groups. So we have used ITS, LSU, and alpha elongation factor, and furthermore genes to check whether these most informative sessions, which are the most informative genes. And we have performed maximum parsimony analysis, we have performed neighbor joinings, we have performed mixed bias analysis, as well as posterior probability. Then we found that the original Aspergillus caricae, it's established as type, is falling across Nothopacellora and Panthospora complex, like group, rather than this particular strain was assembling far from those particular species. Because these are distant cryptic species which are found in having same morphological or at least identical morphological characteristic to that of Aspergillus. Then we decided to put up the Pedro Krausiella, after the legendary mycologist who contributed this whole life, 20 years around, for the Mycospherulia group. Then also we have to propose this epitopification. Why it is necessary? Because if you are putting up an old material, there are protocols to define the nomenclature. So how to do it? You have to establish a material, validate it, authenticate it, compare it to the holotypes, and establish the cultures, make it available in at least 1 or 2 culture collections. Then you should proceed for epitopification. Then Constance Bench, sitting at Myco Bank at Westerditch Institute, if you can contact her, she will also advise you with the procedures how to go ahead with it. Also, you can get support. So when we talk about culture collections and herbaria or fungaria, you can also read about epitopification and the role of herbarium for unknown description of unknown fungi, important mycological and plant herbaria, and tracking the plant killers. This is all about how climate change affects the plant killers, whether diversity present. These are few key papers you can go through and advance your knowledge in this direction. I'm glad to see that even mycology is taking shape into greater heights. Through Fungi Foundation, they have the first country in Chile. They established a conservation policy at the national level and contributed to include fungi in the conservation initiative of a country and established. And this has been very well mentioned in these secret partners, this hunting for fungi, the sustainable global system, and these world recognized systems. Thank you very much. As this is the partnership meet happened in WDCM, WFCC, and I was lucky to represent India there to talk about Indian fungal culture collections and diversity. And now I thank the organizers, MycoAsia, for giving me this opportunity to share a few key informations related to the topic. Thank you. And I am sorry for taking much more time.
Thank you, Raj. This was a very informative talk. I learned a lot about fungaria and how it can strengthen fungal taxonomy and the role of fungal culture collections. And so now I would like to invite the audience for any questions. There was a lot of information to handle for sure.
It was. I'm sorry to pump up with a lot of information because...
No, that's okay. It was insightful. So any questions from the audience?
Hello.
Yes, sir.
Yes, Dr. Kara.
Hi, hello, Rajesh.
Thank you, sir. Yes, sir. Please.
Thank you for delivering a nice lecture.
Thank you, sir.
And as far as the recognized repository are concerned, spread worldwide. So, do you have the inventory of whole words when anybody submit isolates? So, before giving the accession number, is it tally whether this fungus has already been deposited with some accession number or not? The first question. And the second one is that I think more than 20 years this Agarkar Research Institute is running the repository center, and yet till now only submitting 5,000 isolates is not looking less this number.
Okay, and can I answer you?
Yes.
Why not? Yeah. Your first question was depositing your strains and whether it is available across the globe, isn't it?
Yeah.
Actually, if you're part of global catalog of microorganisms and cultures, details were shared with the WDCM, then only it's available there. That's why if you want to become a player of world arena, you have to become partners with greater recognized organizations under the treaties of international guidelines. So coming to the diversity, protection of biodiversity of India is concerned, the number of 5,000, it's a huge number. When you talk about 4 scientists day and night working in a culture collection and preserving and maintaining this culture. Otherwise, if as you are thinking, if you want 40,000, 50,000 culture, we must have such a great facility established by central government of India for even each satellite culture collection we might need, because it's highly costly, and we must need great funding, and we need visionaries. For example, under the guidance of Dr. Sanjay, we could target different groups of fungi, and we are maintaining all these species for supply. And we are also focusing on training these people and preservation of culture, identification services. We are getting around 300 to 500 strains for identification. These are the added burdens of our mycologists who are hardcore mycologists working in the lab. We are also now supplying photographs, descriptions for the people, and also we are mentoring youngsters. We have around 10 to 20 PhDs coming up to take up the modern mycology into the next level. These are the challenges. When we talk about the deposits and the numbers, yes, there are huge challenges resting on the country, and there must be adequate funding to support, as well as expert. Even we have eminent mycologists from different states of India, and they are serving in different societies like Mycological Society of India. We must avail their services, because nobody is perfect and accurate in fungal identification based on morphology. Also, we must need young bloods to take up the next generation sequencing, as well as multi-gene sequencing, phylogenetic analysis, and also some collaborations with international groups. Then only we can lift up this number, as well as quality of research for our country. That's the prime motto of National Fungal Cultural Collection of India. Even MycoAsia, why we collaborated for Asia is for this particular purpose. That means, even different minds, different mindset, different culture, different country across Asia, we are trying to pull up the information, collaborate together. You can see that the last outline of fungi, or even early divergence of fungi, basic basal clades, the leader was Nalin. He was taking up this for Asian countries, because when you talk about Hibbett's classification, none of the Indians were participating in the Hibbett's classification. Now, if you want to talk about the classification of fungi, you can see Dr. Damodara Shenoy there, Dr. Rajesh Kumar there, representing your country. So this kind of progress already happened, and we have to take it into the next level. The much more funding, support from governments, and eminence working in the mycology area. Thank you, sir. I hope I addressed your question.
Yes, we understand. But what is going on these days? Because earlier when we were depositing, then what descriptions, photographs, and structures we were sending, either in Q and IRI, they were accepting and just giving the accession number. Okay, so
yes.
Now the things are changed. You people first identify, take the picture, and everything you people do, and then provide accession number. If someone is sending his or her fungus with photographs, with identification, with ITS sequences, so do you again re-identify, then give accession number, or just based on his or her data, you people give accession number?
Sir, there are ways for doing it. Actually, we prefer to identify every biological resources coming to our center through the standard protocols. But
yeah,
that is the basic criteria for putting up accession numbers to a culture. We have to culture that, revive those strains. What is happening is, some of the times when we get culture, there will be multiple strains mixed in the cultures. So as we have experts, we culture it on different media at different temperature, we differentiate it. In such cases, we report to the party that we have some issues. Otherwise, if they are assured with the standard pattern, for example, if you are submitting a Penicillium aspergillus or Chalanomyces culture, the basic criteria for authentication, not identification. Authentication means it is more macromorphology based, micromorphology based, 4-gene sequencing, concatenation data, and even in some groups, chemical profiling, as you were discussing earlier. Some key metabolites were identified in the group. When such cultures comes, we were glad to admit that we'll accept it and deposit as such with the data available from the depositor. And this is not happening across India because we know that the cost effectiveness of doing such research is huge, because you may not able to deal with such intense research in a normal laboratory. So that is why we come up with the thought process that, we have some facility for sequencing, like not only barcode sequencing, ITS, and ribosomal DNAs like LSU or SSU, rather the secondary genes. You can see that alpha elongation factor, RPB1, which are necessary for fusarium identification. If you send me a culture for deposition of fusarium, I will make you aware that these are the criteria for fusarium identification today, and what would you like to do? So we also try to contribute to the Indian mycology through the available funds. We sequence few cultures on request and through collaborations. There are mode of doing it, and our curator is so generous to build up such a facility that we are available for your service, as well as in a physical manner for deposition identification, as well as intellectual manner. That means we can guide you in getting better identification services.
Okay, Rajesh, thank you for educating us. But we were understanding that repository center has duty to just what I am submitting and what I am giving the elucidation of that fungus, and if just morphologically they can match whether the submitted isolate is that claimed isolates are not. If differing, then they can give their opinion that this fungus is not this and this may be this. So after identification, that will be deposited in the centers, and accession number will be given. If any other scientist want to test whether the claim, whatever the potential or attributes has been claimed by a particular scientist, then he or she may challenge it if they found the claim is wrong. But the repository center has they have to maintain the isolates in what form depositors have submitted. I was under this impression, but you are saying that
can I add?
Have to yeah, you can, but okay.
I think the time is short. We are already 30 minutes.
So, but anyway,
I have a question.
repository centers of India working well, and you people should ask more manpower to work more rigorously, so that wealth of India could be preserved. Okay, thank you very much, Rajesh.
Thank you.
I have, madam, can I come in? I have got a couple of questions for Dr. Rajesh.
One quick question, and then you can contact him on his email.
Yeah. Dr. Rajesh, this is Dr. Dhanurkar again from Symbiosis. I have just two small questions for you. Yes. First thing, apart from lyophilization or liquid paraffin, is there any other promising method for preservation of cultures where cultures can be revived immediately with whatever metabolite production potential it has in its original condition? Second thing, whatever cultures if are deposited at the National Culture Collection Center, if I buy a culture from you for some research purpose, and if I find that has an industrial potential and I can explore it for commercialization, or is there any legal compliances to be followed, or any royalty to be paid to either to the depositor or to the Culture Collection Center if I have to commercialize that culture bought from this Culture Collection Center?
Your first question was on preservation techniques. There are 5 to 6 different preservation methods: active culture forms on glycerol preservation, mineral oil preservation, and even if you know the host and everything, we advise people to go for natural way of preserving after double autoclaving the grains and other things. So some cases in plant pathology, they have to maintain their strains in a particular substrate. Then only they will have particular characteristics to identify. When you are considering your second question related to availing cultures from National Fungal Culture Collections, usually you sign a form with agreement with culture collections. Also, there will not be any royalty as such when you take cultures from general deposits. And if might be there are safe deposits cultures, if there are some accessions for under research studies, there will be more detailed rules and regulations. Even for patent culture, until the patent is published or invalid, it may not be supplied to public. So otherwise, when you wanted it for a larger study, for this is the mode of collaboration method, and we are never denied to deposit anybody across India with a morphological identity to our culture collection. We warmly accept it. We support researchers with different levels of facility available across them. So that is the way it functions. But we try to validate, authenticate, and keep up with the international standards.
No, my question was, if I buy the culture, if I get a culture from culture collection and I find it has a commercial viability potential to explore it for commercial activity or commercial production, maybe some metabolite which is important from industrial point of view.
Okay.
Can that be commercialized? Can that culture be explored for commercial utilization?
Yes, within India you can commercialize, but you cannot supply this to outside India, because
of course indigenous. Yeah, of course indigenous utilization.
Indigenous you can utilize for applications, but you must designate the culture collection, recognize that these cultures are coming from this institute.
Yes. That is absolutely. Any other legalities or any other compliances needs to be done. That is what I just wanted to know.
Yes, the basic compliance is with dealing with the culture collections curators.
Okay. So I would like to, sorry to interrupt you guys. You can, I request Dr. Rajesh to please drop your email ID in the chat box, because there are a lot of people willing to ask you more questions, and we really don't have time right now. So I'm sorry for that, and please continue over email. Okay, so Dr. Shenoy.
I just want to make a comment about the commercialization of the culture retrieved from accessed from a national fungal culture collection. If you develop a successful technology, you will have to give the credit to the original contributor. That is for sure. Tomorrow he can sue you for
infringing
his property right. Okay, and thank you, Gunjan, and we can move
for clarifications, Doctor. Appreciate it. Thank you all for giving me the opportunity. Thank you, Gunjan. I'm not taking much time. Take care.
Thank you, Rajesh. And now, without any further delay, I would like to welcome our new next lecturer, the speaker. He's Professor Udeni Jayalal. He's working as the head and professor at the Department of Natural Resources, Sabaragamuwa University of Sri Lanka. I hope it was correct. He is the first PhD holder in lichenology in Sri Lanka. And today he'll be talking about the lichen research in Sri Lanka. I'm sure it will be very exciting. So over to you, Dr. Jayalal.
Yeah, thank you, Dr. Sharma, and thank you very much, Dr. Shenoy and all the organizing committee to giving this opportunity. Good afternoon, all of you, and warmly welcome for this special event. Right, now all the other fungi, when compared with the lichen, it is somewhat different from the other normal fungi species, right? As you know, the lichen, it is a combination of fungi and algae, or sometimes we can see some cyanobacteria rather than algae. Next slide, please. Go to the other one. Yeah, actually, before I start this talk, I need to pay your attention for somewhat recently findings related to the lichens, right? So far, we know that the lichen is a combination of only a fungal species and the algae. But recently, some scientists found that rather than a single species, sometimes in some lichens, actually, it represented several fungal species, right? So that it's really, difficult to synthesize such lichen in the laboratory scale. I will explain some of the examples. So this is one of the recent finding that some scientists found that apart from the one algal, the fungi species, and they have found another yeast species that is incorporate to form the lichen species. Next slide, please. And this is another example of having two or more fungal species come together and contact with the algae species, and where they produce the lichen species. Now, one example is this one. You can see the Tremella vulpina is the lichen species, but when considering their free living state, some of the fungi, those belongs to the same genera, also come to contact with the same lichen, and finally they form the final stage of the lichen. Next slide, please. And this is the one actually I just wanted to mention. Now, considering the lichen, actually many scientists were trying to remake or synthesize the lichen inside the laboratory, but unfortunately many of the attempts have failed. But you can see in the left hand side of your screen, here you can see the resynthesized thallus, and when the same lichen, if it is present in the natural environment, you can see the difference, right? So morphologically it is completely different from the synthesized one and the natural one. But very interesting thing is that when compare the secondary metabolites of lichen, actually it is almost complete, or almost same, or the 100% similar to the resynthesized lichen thallus as well as the naturally available lichen. So it is make some good evidence that if somebody needs to extract any secondary metabolites from the specific lichen, so rather than going for the natural lichen, so we can resynthesize them at the laboratory, and after that we can extract what are the chemicals that you want. Next one, please. And this is the other recent finding, that that is the so far people or the scientists believe that if any lichen especially present on a bark, right, they believe that there is no any special relationship. I mean, there is no any absorption process taken place by the lichen when compared to their host species. But recently scientists found that actually some fungi hyphae just grow inside the bark or the stem of the host species, and they just extend their fungal hyphae into their xylem and phloem, and after that what they do is sometimes they excrete some chemicals, and at the same time they absorb the requirement that they need from their host species. So that is very important, because we have to think that whenever you see any lichen species, either on a rock species or any other plant species or whatever, now we have some idea that they not use it as a host or a place to live, but they have some interactions between their host species and the lichen species. Can you go to the next one? Now, considering the worldwide scale, so far around 20,000 lichen species identified, and many of them belongs to the Ascomycota. And apart from that, very limited or the very little amount of lichens were identified from the Basidiomycota. Now, when we go through the species generic level to the classes level, now you can see many of the fungi that responsible to make lichen species belongs to the Ascomycota. Next slide, please. And this is the another important point. Now, if you go through this graph, actually many of the lichens were discovered from 1976 to 2015, or the current date, let's say 2021. So we call that the global taxonomic warming, because if we compare the number of lichen species that scientists have been found before 1970s, it was kind of the little amount of lichens, but half of the currently accepted genera actually found during very recent time. Next slide, please. Considering the diversity within the lichen, actually there are many families available. Among these families, many species, around more than 2,000 species, belong to the family Paramageetham, and similarly, somewhat close to that number represented by the family Grapidaceae. So that you can see this type of genera, we can say those are kind of ultra diverse genera. And when go to the ultra diverse families, when go to the generic level, again you can see how this different genera represent this type of the different number of lichen species. But considering the Graphis species, this is hyperdiverse actually. Considering Sri Lanka, I will explain later. Many of the studies actually recently completed related to the genus Graphis. Can you next slide, please? When considering the global lichen red listing project that is done by IUCN, and 50 lichens are so far nominated for red listing in 2017, and 87 are nominated for 2018. But considering the Sri Lankan lichen, actually none of the Sri Lankan lichens are listed for the global lichen red listing project. Next slide, please. So these are some of the example. Now you can see the diversity and beauty of the lichens. Actually, it was published in 2017 by Lucking et al. Next slide, please. So this is the another interesting one. So when you look at this, somebody can get some idea that some are kind of mushroom, but actually these are some of the lichen species. Actually, those belongs to Basidiomycota. Next slide, please. So considering the highest lichen diversity spot on Earth, actually very recently, based on the high number of research work and the people that wanted to work on the lichens, now I mean, India, Sri Lanka, and all the tropical countries having the highest diversity of lichen on the earth. But early days, it was in the other countries, because like temperate countries, because there were many scientists and they were worked on the lichen diversity and lichen taxonomy and other purposes of the lichen related work. Next slide, please. Considering my country, you know that where it situated, right? Actually, my country is, I mean, 1/60th smaller than India. However, when we compare the lichen diversity, actually, we are having a high number of lichen diversity when compared with the other countries. Next slide, please. Now, considering my country, actually, we have different floristic region, so that actually we have different climatic regions as well. So that I mean, just go ahead. And based on these reasons, actually, I think we have that much of large diversity of lichens. I mean, when considering the other plants, not only the lichen, but also considering the higher plants also, Sri Lanka having high diversity when compared with the other countries. Next slide, please. Next slide, please. Right. When actually considering the lichenological studies in Sri Lanka, if you go through this list of the scientists, those who have been working on lichen, actually many of the scientists were the foreign scientists, but in 1999 actually we had a lichen workshop. That was the first lichen workshop in Sri Lanka. And at that time, the participants and all the other resource persons have collected many lichens. And based on their findings, actually, what they predict that probably Sri Lanka has more than 1,000 lichen species. And when compare with my country and the other countries, actually you can see in Sri Lanka we have only limited number of research groups who are working on the lichen. Myself and Dr. Gautami Veerakonda. Now she is the curator of the Natural History Museum in London. She also working on lichen. And considering the India, actually, I think India have more than 2,500 lichens, and there are many more research groups who are working on lichen. But the same thing, if we compare with the other countries like British Isles, you can see there are a large number of people who are working on the lichen. So that's the reason. One reason that earlier I mentioned that in the tropical region there were many lichen record during the past time. Next slide, please. So what are the main objective of Sri Lankan lichen research? One is to document lichen in undisturbed forest types and disturbed habitats, because considering Sri Lanka, actually we have completed less than 5% of the total area of the country. So that there is a huge area that we need to complete to survey on lichens. And the other one is to determine differences in lichen assemblages and their association with microclimatic and phenotype variables. And the other one to identify potential indicator species for vegetation types and disturbances level. Even we have completed some of the recent works related to identification of indicator species. Actually, I am happy to say that during our works, we were able to find several indicator species, especially for the air pollutant indicators. And the other one is to identify renewable source of bioactive natural products from endolichenic fungi. Actually, not only the endolichenic fungi, nowadays we are working on lichenicolous fungi and also lichen forming fungi, also another area where we are working. We know that based on our recent findings, we were able to find so many bioactive compounds from Sri Lankan lichens. So that is nowadays, apart from the taxonomic work, we are mainly focusing on some of the applications, like how to identify bioactive compounds and how to isolate them and so on. Next slide, please. Considering the lichen checklist, actually, as I mentioned, we have completed around 5% of the land area. And during that study, we were able to collect more than 1,000 species. And recently, Dr. Gautami and others describe actually many number of new species and around 1,000 or somewhat close to the 1,000 new records. And also there were 140 new records in Indian subcontinents, and there were more than 20 new records for the Asia. Next slide, please. So now Sri Lanka is now become the country with the highest number of new species and records per unit area, and estimated number of lichens of Sri Lanka should be more than 5,000. So you may feel why we have such diversity. So what we believe that it is a complex earth movement have resulted in remarkably unique lichen biota with high levels of local endemism. Next slide, please. And distribution and biogeographical relationship of Sri Lankan lichens. You know, the majority of Sri Lankan lichens are gondwanic species, and some distribution patterns are very ancient, that reflecting past land connection and the fact that lichens evolve very slowly. And majority of lichens have their closest relatives in in the Himalayas, then South India, Africa, and Southeast Asia. And new species have, few species have some relatives in alpine and polar regions, Europe and North America as well. Next slide, please. So this is the latest publication. Actually, we were able to make the very recent lichen checklist, and it was published in 2020 in the National Red List of Sri Lanka. Next slide, please. And where we were able to identify around 876 species in 233 genera, and those were in 60 families. Next slide, please. So this is the one I mentioned, and the interesting thing is that considering this 876 species, actually there were more than 150 species are endemic to Sri Lanka. Next slide, please. So these are some of the findings. We were able to describe these different species new to science. Next one. So these are some of the photographs of such crustose lichens. Next one, please. Next one. Yeah, this is another article. Next one, please. Yes, now considering Sri Lanka, actually these are the places so far I have mentioned where we have completed the survey, right? And you see that what is the remaining area, that it is a very large area where we need to complete on lichen survey. Next one, please. So with that, I will show you some of the examples of the research work I have carried out with my undergraduate students. Actually, usually that students are given only 3 months period. So during that short time period, actually, they were able to identify, or they were able to publish many articles. So this is one of the interesting one. We have collected some of the lichen, and we just checked their antibacterial activity against human pathogens. Next slide, please. So actually, we got very interesting results. Still, we are working on them. Actually, why is that? We just use the crude extract, and now we just wanted to do further works to extract this unique chemicals, in which chemical that shows this type of antibacterial activity. Next slide, please. This is also the same one. Next one, please. Next one. And also we have done another one related to the antioxidant, anti-diabetic and phytotoxic properties of locally common native lichen species, somewhat close to my university. Next one, please. During that work also, actually, we were able to find very interesting results. That mean many of the lichens that we have collected from the university area having very good results. That mean high antioxidant level and some other results. Actually, the positive results we obtained during our study. Next one, please. Next one. And the next one. And this is one of the picture where you can see how this lichen extract or the secondary metabolites show root inhibitory activity against some of the plant species. Next one, please. And this is another example. Actually, we have completed lichen diversity survey along the ambient air quality in a gold district. Next one, please. And we have fitted this type of passive air samplers to monitor the sulfur dioxide, then nitrogen dioxide at the atmosphere. Next one, please. And at the same time, we have collected the lichen species from where we have placed that specific instrument. Next one, please. So you can see when compare the lichen diversity and the nitrogen dioxide concentration, somewhat the lichens and nitrogen dioxide very close to the city area. You can see in the red color, actually in that area having less number of lichen diversity and high nitrogen dioxide concentration. And when we go further away from the city area, from suburban to urban areas, you can see the how these diverse, that lichen diversity and the nitrogen dioxide concentration. Yeah, so this is the same one, how this lichen diversity change with the sulfur dioxide concentration. Next one, please. Yeah, these are some of the values we obtained. Next one, please. The same one. Next one, please. The sensitivity to atmospheric pollution, climate change, and habitat modification is very important, right? Therefore, lichen are threatened by large-scale destruction of natural habitats over the past century. That is not only common to Sri Lanka, but also it is common for the other countries. Now, actually, we are mainly focusing how to conserve the lichen species in Sri Lanka. So nowadays we are planning to do something to do that as well. So next slide, please. So with that, I will conclude my presentation with the statement given by the Mahatma Gandhi. What he says, Earth provides enough to satisfy every man's need, but not every man's greed. Thank you very much.
Thank you, Professor Jayalal. It was a lovely talk. So now, audience, do you have any questions? So there's one question.
Hello.
Are there invasive lichen species in Sri Lanka? And if so, what impact do they have on the local environment?
Yeah, many of them are in the mountain area. I mean that may be a reason that area having enough humid condition and the low temperature and enough sunlight also there. So that might be the reason that we are having high lichen diversity in actually somewhat wet mountain area.
Okay.
Hello.
Yes, Yogesh.
Hello, Dr. Jayalal. How are you? Fine?
Hi, I'm fine, thanks.
Just a query that as far as India is considered, that we have the coastal area full of sands. It's not rocky. So in your country, if you have a rocky seashore, then whether you have gone for maritime lichens or not?
Yogesh, it's a very interesting question. Actually, we were trying to find some lichens in that seashore.
Okay. And what about the, as you mentioned that some lichenicolous fungi you have told that, could you share with me that whether you have find some lichenicolous fungi from that region? Because
yeah,
as
sure.
far as
Actually, yeah, now actually we just started to work on that, Yogesh. So I will share with the findings later on. Now, considering your questions, actually we have found only the Roxella montagni only on the coconut plants. So that is the most abundant species somewhat near to the coastal area.
Okay. So don't you have the rocky seashore there? If you have rocky seashore, then maybe Kalaplaka and Valucaria.
Yeah, we have, but actually we couldn't find any lichen species on them.
Okay.
Yeah.
Okay. Then I would be having very much interest if you could share this lichenicolous literature with me.
Yeah,
if
sure,
you have.
definitely. Yeah, definitely.
Thank you very much. Thank you.
Yes, Dr. Raj has a question.
Thank you, Udeni Jayalal sir, for enlightening us and being a part of MycoAsia. My question is related to the diversity of Sri Lankan lichen in correlation with the Western Ghats, as it is considered to be a mega diversity hotspots, whether there is any link you found common to both these areas. The second
Yes.
question,
Yes.
the second is an additional question. It's about what is your take on the molecular data and its phylogenetic positions, considering the Western Ghats and Sri Lankan lichens?
Actually, the considering the Western Ghats lichens, many of the lichen represented in both areas. Now I have been working with South Swaminathan Institute, where some of the researchers are working on them. So I mean, there is no much difference between Western Ghat lichens and the Sri Lankan lichens. So I mean, that is common for plant species as well. Considering molecular level, actually, we haven't compared any molecular characteristics with Sri Lankan lichen and Western Ghats lichen. So actually, your question is very important. So I'll try to do something related to your question.
There is one more question from the audience in the chat box. Dr. Seema has written a very nice session. Can you quote any clinical research on Selaginella species?
I couldn't get Sharma your question.
Can you quote any clinical research on Selaginella species?
No. We haven't done any.
And there's one more question. The diversity of lichens correlates with the diversity of photobionts in them, or they differ?
No, actually, considering the photobiont diversity, we couldn't find such a diversity with the photobionts actually. That means many lichens, although we found different lichens, many of them are having the same photobiont, most of the cases. The only difference is the fungi species. So I mean, it is not common for our lichens, but it is common for some other lichens as well. So that's why many of the scientists are not much worried about the diversity of the photobiont.
Okay, so there are two more questions.
Madam, I have one question.
Okay, please
Hello.
ask.
Yes.
Sir, in one slide you have shown when we culture this lichen, it shows a difference when it is compared with a naturally, you showed Asnia species. How come the cultured lichen, how come it will show difference? Because definitely it should not, because now when we culture also the same lichen appearance, it should be there. That's what my question.
Thank you, Mr. Rajan, and your question. Now, actually, at the beginning, now when we culture in such lichens, at the beginning, they just try to start as the normal thallus or the natural thallus, but the differentiations will start with the time. Once we keep it for a long period of time in the culture media, then they just started to change their morphological characters. So that is the common problem that many scientists are facing. I don't know whether you got
because here in culture, do we need to add both? Because this lichen is a combination of algae and fungi. When it comes for culture, do we add both the algal and fungal inoculum, or do we need to add? Because that's what it happens. Both the inoculum we need.
No, the issue is this actually, as I mentioned at the beginning of this presentation, there are some hidden organisms are available in the lichen formation. Now, actually, we don't know what is their duty to formation of the lichens. Now, although we believe that there is only one lichen forming fungi, but inside the lichen, there are some endolichenous fungi as well. So sometimes I mean, some that endolyconic fungi may have affected for the formation of the lichen. So actually, we don't know the actual situation happening when they are in a natural environment. So if we know what is happening in the natural environment, or if we can provide the same situation in the laboratory level, then most probably we could get the actual I mean thallus that 100% looks like a natural thallus.
Are you trying to isolate any lichen, sir, in your lab?
No, I mean, I have been working not in my country, but I have been working some other countries where I have worked on such trials, but I was unable to get the proper lichen thallus when they grow, when we try to grow them in the laboratory.
Okay, okay. Thank you so much, sir.
Okay.
So good afternoon, everyone. So I would like to welcome all of you for the second technical session of the lecture series. So as you know, this is a joint collaborative work from us, from MycoAsia and this reputed university from India. So our in the first session, we had 5 presentations, talks from some good mycologists, eminent mycologists. So now we have the second session, technical session. So as the first invited speaker, we have Dr. Rohit Sharma. So Dr. Rohit Sharma obtained his PhD from the Department of Biological Sciences, Rani Durgati University, Jabalpur, and he has worked as a curator at National Center of Microbial Resources, Pune. Currently, he's working as a professor and R&D coordinator at SVUniversity University, Bhopal. So, Dr. Rohit Sharma, I would like to invite you to deliver your talk.
Yeah, thank you, Doctor. Good afternoon, audience. My apologies, first of all, because due to some urgent travel plans, I have to give this talk, as you can see, in a car. So, by the time Dr. Shenoy shares this slide, I'll just try to introduce the topic on which I'll be speaking. That is the development of efficient and low-cost biotechnology for color removal of biomethonated spent wash from distilleries. So basically, yeah. Thank you, Dr. Shenoy. Yeah. So basically, the distilleries, they are producing a lot of waste that we call as a spent wash. And basically, as you must be aware that the distilleries, most of the distilleries, they are associated with the sugar industries, and sugar industries, whatever molasses they produce, and that has been taken into the distilleries for the production of the alcohol. And the most of the Indian, in fact, all the Indian distilleries are based on the sugarcane bagasse and the sugarcane molasses that is obtained, that's based on the sugarcane industries. So till now, there is no such biotechnology available, or the technology of the color removal available for the distillery waste. And as we are growing, the CPCB, the Central Pollution Control Board, has repeatedly made stringent norms for the waste treatment kind of thing. And that is why it has become more relevant in present time that we develop a kind of biotechnology which can remove the color of the spent wash. So a spent wash basically is made up of it's a brown color. I'll just show you some photographs. It's a brown color, and it is made up of several kind of compounds. And the Indian industries does not have any technology. Basically, what they do at present is they are not having any treatment for the waste that is produced by the distilleries. So, and next slide, please, Dr. Shenoy. Yeah. So when we did the physicochemical analysis of the spent wash, it was having a very high COD and BOD. That is the only thing that is were in the parameters. Apart from that, there is no treatment for the total solid and the color. That is a very brown color. The right hand side, on the right hand side, if you can see, these are the pre-methanated. The above photograph is a pre-methanated of a distillery of the Maharashtra, and the below is the post-methanated. The pre-methanated means the distillery waste comes directly outside. It is around 70 to 80 degrees of temperature. It's a little bit warm, hot. And the some of the distilleries have started doing the kind of anaerobic digestion of this waste, and then they release it after post-methanated. That is, after they produce the anaerobic digester, they have the electricity produced, and that biogas that's produced, and they produce the electricity out of it. But the color still remains the same both in the pre and the post distillery waste, spent wash. And this spent wash is basically made up of this caramel is there, melanoidin is there, and several other phenolic compounds are there. And the prominent one, that is the brown color that is you are seeing, is basically because of the melanoidin. This melanoidin basically is formed when the charring happens, and the sugar and amino acids that they char, and they produce melanoidin. It is the literature shows that it is harmful for both humans as well as the crops. In some of the regions in Telangana and Andhra Pradesh, even the groundwater has been contaminated, and when the borewells, and they are releasing the brown water out of it also. So just to tell you the magnitude of the problem in India, there is around more than 450 distilleries are there in India, and in Maharashtra itself there are more than 150 to 200 distilleries are there. And to produce a single liter of alcohol, they use around 10 to 14 liters of pure water. That is pure water they are using for that, and that amounts to around 4 lakh liters of pure water for distilleries are using per day. So that is the magnitude of the water that their consumption. So with the the underground water is becoming a scarcity nowadays because of several factors that we are all aware about and we know. So that's why we intended to work towards the kind of distillery that is there. Based on the next slide, please. So some of the distilleries, they are also storing them as lagoons. So they prepare these lagoons, but these are not proper scientifically lagoons. That is the cemented walls, nothing is there. They are only in the ground, and that is the seepage happens, and then the soil and water is also contaminated. And many of the industries even discharge this distillery waste in the rainy season along with the muddy water, and it flows. So they do not have that kind of treatment facility till now. And the amount of waste that is generated, you can understand. And the lower photographs, if you can see, they are also spraying this distillery waste with the help, with the kind of they are preparing kind of fertilizer, biofertilizer, using this waste also, and these are also being distributed to the farmers for that. And next slide, please. So at National Center for Microbial Resources, where I was working earlier till last year, so there we had a huge resource of microbial resources, so which were available to us for the screening of the screening for this purpose. And this was a Department of Biotechnology funded project, and where they asked us to screen all the microbes for the this purpose. So what we did is we screened about 10,000 of microbial cultures along with both bacteria and fungi. The problem with the bacterial cultures was there that they were not showing that much of decolorization, number one, and number two, those cultures which were showing the decolorization during the re-fermentation, they were losing the properties of the decolorization ability, and that's why we could not take those cultures further. However, the fungi was quite promising. So hereafter, the whole project were focused on the fungi and the distillery waste, that is the spent wash. So apart from the culture that we already had, we also tried to isolate, based on the literature, we tried to isolate, did some surveys near and about Pune forest and the Mulshi forest and Tamni Ghats and other areas nearby the Pune, where we tried to collect wood rotting fungi. Because as per the literature, the wood rotting fungi, that is both white rot and brown rot, specifically white rot had the more potential. So we tried to screen collected those kind of fungi, cultured it and then tried to screen those fungi also. And on the right hand side, if you can see, I have mentioned a table kind of 1 star, 2 star, 3 star. So we categorize these data based on the kind of potential they were showing. So 1 star means that is the very least potential, 2 star means little bit better potential, and 3 star means which is showing the highest decolorization ability for decolorizing the spent wash. So we found around 3 cultures were found to be having a 3-star activities. All were fungal cultures that we took for the secondary screening. Next slide, please. So this is the data that you can see that some of the strain even showed more than 80% decolorization ability into it. So this secondary screening we did in the around 500 ml to 50 ml flasks, wherein we tried to have around 150 in 250 ml flask. We tried to have 150 ml of the distilled spent wash, and then we inoculated our culture. The initial time, all the cultures were then 3 cultures were there, and the second screening in fungi also 1 culture lost their ability, and there were only 2 cultures, that is PF13 and PF09, that were taken further for further studies and those things. So here the number of days it is taking to decolorize also mattered, because we have to consider that we are trying to develop a technology for a industry. So there the retention time is more important, because if we say my culture, my technology will take 15 days to decolorize, so to hold 15 days of waste for a distillery industry is a big task and huge responsibility and a huge kind of thing. So that's why we also wanted to reduce the time also. So next slide, please. So if you can see here, this is the 0 day, 10 day and the 15 day thing that we did. So here we did 2 things. First, we did normal up to 15 day, it was decolorizing, and you can see the amount of decolorization that it was showing. It has a lot of decolorization it showed. Then in the second cycle, what we did is, instead of re-inoculating the culture, why I did 10 days? Because the the growth, the cultures were also taking time to grow. In the second cycle, what we did is we filtered the already grown culture, that is the last flask that you are seeing, and the same culture we reused it. So here since the culture were already grown, the time reduced to around 7 days. So the time that was taking to 15 days to decolorize, that reduced to around 7 days for decolorization, because the culture it was grown. So this thing we repeated twice, and we wanted to we are still working, so how many days we can take it further. So this we have done up to twice cycle we were able to do, and we increased the amount of that is this distilled spent was up to that is 500 ml, and we also took up to the 1 liter. So in the laboratory we were able to develop up to 1 liter decolorization, that thing. Next slide, please. So some of the cultures that were not showing, that is they did not did very well, and that you can see that they did not showed in the good decolorization even if in the secondary screening when we increased the amount of the distillery waste. Another point was that we could use only 50% of the distillery waste. We cannot use the 100%, because 100% it was not showing any growth. We started with 25%, we reached up to 50%, and then later on the studies were done up to the 50% of the distillery waste. Next slide, please. So these were able to do up to this. So what we did in the laboratory was that we started from the distillery waste, we inoculated our culture, we decolorized up to it, and then apart from that we also used 2 kinds of thing. We also integrated UV light, that is the physico-chemical method, also we integrated to enhance the that is the decolorization capacity, because UV light is known to degrade melanoidin compounds and break down. So we did 3 kinds of study. First we did the UV first, then chemical, that is hydrogen peroxide 1.01%, then the microbial, and other way also around we try to did it, that is the first microbial, then UV, and that thing. And we found that if we can treat first with the microbial culture and then we give the UV treatment, the efficiency was much better as compared to doing the vice versa. So the enhancement of the that is the decoloration increased by 10% by using the UV light. That means the microbial culture was showing 80% of decoloration, and when we additionally use the UV light, it went up to the 90% of the decoloration using these fungi. And both these strains were belong to the Bacillus mycota, and we are now developing different for the patent that we require different kind of data for in proper we are regenerating and revisiting that data for those purposes, and both are the wood rotting fungi that we found. And the standard that we were comparing was the phanerochaete chrysosporium, because phanerochaete chrysosporium was known based on literature to have the decolorization ability for this spent wash studies. Yeah, next slide please. So we also, based on the literature, what we found that most of the leakage enzymes are known to being associated with the decoloration ability of the fungi. So we also tried to screen whether they have the ability to produce the leakage enzyme, and we found that all the two strains were having a good amount of leakage producing ability by using the ABTS as a substrate. And the plate well assay as well as the spectrophotometry. So what came out of it was we were going in the right track, that okay, these are the wood rotting fungi, white rot fungi. Then second is they have the leakage producing ability. Next slide please. So we also tried to do the genome sequencing, and we are studying the data at present, analyzing the data, and what has been found coming out of it is that these leakage enzymes are associated with the a class of enzyme, that is the oxidoreductases enzymes. And we found that both these strains, that is the PF9 as well as PF13, both had a high amount of high number of scaffolds for the oxidoreductases, hydrolases, and transferases. And all these enzymes are known for the leakage kind of group of belong to it, and that's why we found that okay, this is can be associated with it. Why we are doing the genome sequencing? Not just for the sake of doing it, but what we wanted to know is that okay, it is degrading, it is the basically trying to decolorize the thing, but what we wanted to know is that what pathway it is following for the degradation, whether it is what kind of pathway the fungi is taking, whether it is the leakage, because it is still not clearly known whether the leakage is being the sole responsible for the declaration of the spent pore. So we also wanted to know what kind of pathway it is the fungi are following for it. Next slide please. Yeah, one more. Yeah. So based on the recommendation of the project committee, we had modified our objective also, and it was clear that okay, the spent wash is getting decolorized, and the role of fungi is there. But to establish the concept more, the committee has suggested if you can purify the melanoidin, it is decolorizing spent wash, that is okay, but whether it is degrading the melanoidin or not, we wanted to know. So we purified the melanoidin from the spent wash, and then we showed it the degradation, inoculated our fungi, and then we analyzed the data in the HPLC. Still, this thing is being done again. We are trying to repeat it, but the preliminary data showed that the first, as you can see in the first diagram, the peak that was for the melanoidin, that is not there in the treated that thing, and a new compound is getting formed. So now we are know that the melanoidin is getting degraded, that is true, but the other compound that is formed, whether it is toxic or it is less toxic than the melanoidin, or whether it is serving our purpose or not, that we are trying to work on it. We are again trying to repeat these our experiments on the HPLC and these things, so that we can confirm that yes, melanoidin is degrading and a new compound is formed. And what is this compound? We are trying to identify it. And we are hopeful that it should be not more toxic than the melanoidin and the harm that melanoidin is doing it. So the aim that we are looking at present is that at least if we are able to reuse that 4 lakh liter of water by the distilleries, then also our purpose of the biotechnology will be solved. We do not want that, okay, the water will be so pure that it can be drinkable, it can be released in the aquatic environment, and it won't disturb the aquatic environment. Even if it is not there, even if it becomes potable for reusing in the distillery industry, we'll be saving 4 lakh liters of pure water per day if this technology we are able to develop and commercialize. Commercial means up to the thing. So we have developed in the laboratory. So what we have done in the laboratory is that the distilled waste we put in a chamber, it is taken up in the container. There microbial growth culture is there for 10 days or 7 to 10 days, and from there it is filtered out, and then this spent water that is treated is again exposed to UV light for around 1 hour to 1.5 hours at a particular kind of distance that we had worked out, and then we found that it can be discharged kind of thing. So this working model we have established. So where we are at present, the DBT has asked us to collaborate with some of the industries who are into this, like maybe Piraz or some other industries who are into the either in the fermenter kind of thing or bioethanol or kind of thing. So who can help us to extrapolate? You see, we have reached up to 1 liter. I am a mycologist and I am working in the laboratory. I do not have the expertise to take this thing into a 40,000 liter of fermenter that is out there in the distillery plant. So we require industry person, we require industry collaboration to take this thing and extrapolate this data and take this to the that level. And it needs 2 to 3 years of trials before the technology we can commit, okay, yes, it is feasible and it can be used. So we are at the stage that now this technology should be trialed at the industrial level. So some of the distilleries of Maharashtra have given their consent for the trials kind of thing, means in principle, and now things need to be worked out along with the private industry to take this up to that level. Next slide, please. Yeah, if you can see that the data that we had analyzed post-treatment, the BOD COD that was around 70,000 or 84,000 has reduced drastically, and a lot has been around 7,000 or 11,000 it has reduced to that level. And if you can see the lower point, that the color has almost become nil in the post treatment kind of thing. So that is the main we were working. We were actually, COD and BOD is an additional because many other technology have been working on it, but our main focus was on the color. So we were targeting color, and that we were able to reduce to the negligible amount that thing is there. Yeah, next slide, please. Yeah, so we are at the validation of the results of the HPLC. Number one, we are at the we want to scale up this thing up to the industry level. With definitely we need to collaborate with some of the industry because of the obvious reasons I just spoke on. And field trials are very important before anything can be committed on this technology. But being a part of this kind of thing, I am hopeful that this is workable thing. Yeah. The results are promising. Yeah. And next slide, please. Yeah. Thank you so much. Thank you so much for the organizers and to DBT for funding the work, and to the distilleries Maharashtra who gave us the permission to do this work. Yeah. Thank you.
Now the platform is open for questions. Any questions?
Yeah. What
Dr. Sharma, can you come back,
since Dr. Sharma has screened more than 10,000 strains and shortlisted 2 or 3 strains with good traits, he can also try for biopulping with 3 or 4 shortlisted strains.
Yes. Yeah. Yeah. Yeah, yeah, I think that's a good suggestion. I think yes, biopulping is a good idea. I think we'll definitely take up this session and like to work on it, because earlier we wanted to have there was an industry nearby Pune. We approached them, but they did not give us the permission and those things because they were hesitant to share kind of thing. So definitely we'll try to approach some industry, and if they can give us the permission to use it. Yeah.
Thank
you. Thank you, Dr. Sarpangaru. Dr. Samir Damare, please ask your question.
Yeah, Rohit, this is Samir here.
Yes, good afternoon.
Yeah, Rohit, have you done any toxicity studies after all the activities are done?
No, we are in process of doing this for the compound that is now being broken, or you are saying for the melanoidin or the new compound that is being formed?
The new compounds which have been formed.
No, that's what we want, that we are trying to study on the toxicity aspect, so that we come to know whether it is having a more drastic effect than melanoidin, or it is really serving our purpose. So that we have to do it.
Yeah, you'll have to treat with caution, because many a times the broken compounds are more toxic. So that needs to be done first because of the low molecular weight nature.
Sure. We'll take up this.
Yeah.
Thank you, Dr. Rohit Sharma, for your time and your excellent presentation. It has been a very pleasure listening to your work, and the way you explained it to us, with the conviction with which you have shared your experience. Thank you very much. Thank you so much, and I please proceed with your work. Thank you so much.
Thank you so much.
Now we have Dr. Samir Damare as the next speaker. Let me give a brief introduction about Dr. Samir Damare. Dr. Samir Damare is originally from a place called Kankoli on the west coast of India. Then he did his BSc from Wellington College in Sangli, which is in the state of Maharashtra in India. Then he worked for industry for some years, I guess. I believe, yeah. Then he did his master's in microbiology from Goa University. Then he joined for his PhD at NIO under Dr. Rohit Kumar, where he worked on deep sea fungi, biotechnology of it, diversity of it. Then he went to Germany for his postdoctoral work. He came back to India in 2011, joined the National Institute of Oceanography, Goa, Goa in 2011. Since then, he has been serving the organization at a different hierarchical level. He's in the committees. He's on the committees of Shivaji University, Kolhapur. He's also associated with Mumbai University. He's working as editor for many journals, and he's one of the gurus of marine fungi in India, I would say, especially deep sea fungi. He is the guru of deep sea fungi. It is a great pleasure to listen. It will be a great pleasure to listening to him because last year he spent some 90 days exploring different parts of Indian Ocean with our research vessels, Sindhu Sadhana. Over to you, Dr. Samir Damare.
Yeah, thank you, Dr. Shenoy. Wish everybody a happy World Fungus Day. My slides are visible. I am audible.
I think you have to go to the full slide.
Yeah, I'm trying to do.
Okay, it's fine now. It's fine now. Yeah.
Fine. So, yeah, we have been listening to different kinds of talks since morning for this World Fungus Day. And let me take you to the oceans for the fungi from the Indian Ocean. I've tried to cover basically with regards to the diversity and the development of marine fungi and how the deep sea fungal's work has progressed. I have avoided the biotechnological aspect as such with the time constraint in view. So, yeah, this is to give you an idea about the schematic representation of the evolutionary timeline related to fungi. There are many authors for this who are saying like fungi originated here and there, but so far we have no clue whether it is really from the land or if it is from the oceans or the water. But this is the time when you can see 542 to 488 million years ago, somewhere fungi colonized the land. And that is based on the molecular dating data which is available now with us. Yeah, this is these are what the most accepted timeline which is present at present for the fungal presence, which is recorded from the Cambrian to the Ordovician periods. So coming to the oceans, which we have been working like all these years. From school days, we have been hearing that oceans cover 70% of the Earth's, and there are Pacific is the largest, Atlantic, Indian Ocean. This is the one which is closest to us, where we have been working for past 2 decades now, looking into the different types of fungi which are present. So yeah, not only fungi, it said that life originated from the sea more than 3,000 million years ago. This is one of the greatest biodiversity with more than 300,000 described species of plants and animals. And if you talk in terms of diversity, it's really rich with regards to like some places, 1,000 species being recorded per square meter. Now this marine environment, oceans are one of the parts. The second is the estuaries, which is really like a heaven for all the mycologists, where you get different kinds of fungi, and any leaf you pick up, you are sure to get at least 5 to 6 different kinds of fungi on that. And being at Goa, it's really a best place to work with not only the estuaries but the mangroves which are present on the Goan coastlines. So coming to the marine fungi, they have been described as obligate or the facultative ones. The obligate fungi are the ones which complete their life cycle completely in the marine environment, whereas the facultative ones are the ones which can survive in the marine environments, and they are also able to grow in the terrestrial environments. So this is the classical definition given by Colmere and Colmere way back in 1979, and it's being continued and accepted even now. Coming to the marine fungi timeline, 1964 was the first report of the marine fungi isolated from a depth of 4,450 meters by a scientist named Roth. Then in 79 was the time when Colmere and Colbert started working extensively on these kinds of fungi, wherein they managed to isolate obligate marine fungus from wood panels immersed in seawaters at different depths from 5,000 to 3,000 meters. And that is where people started observing and looking at the fungi which are present in the marine environment. But then, even then, they were not easily accepted. There were still a lot of people saying that are they really present, or most of the time they are blown away from the land, and then that is how they are able to colonize. And there were many aspects because of which they were not easily cultivable. One of them was the pressure, and that is where in 1992, Lorenz and Molitoris, they managed to devise an instrument which was used for high pressure cultivation of marine fungi. And that is where really revolutionary work started with regards to, because unless until you have a organism in the culturable or in the laboratory state, you really cannot do much. You have all kinds of techniques to explore them in their natural habitats, but ultimately you need to have a culture in your laboratory. Then only, like best example is E. coli. I mean, there is no process left of E. coli which is really not explored. That is because it is very easy to cultivate it in the laboratory. That is where, when this pressure cultivation chamber came into picture, more and more people started working. And one amongst them were my guides, Dr. Rohit Kumar, where they report from deep sea fungi in 1992. And then in 1998, they were the first ones to publish a detailed study on deep sea fungi from fungi isolated from calcareous shells in Bay of Bengal from around 965 meters. But then again, that was not the so-called deep sea what people are looking into. And 2006 was the one of our first papers. This was from my doctoral work, where we first time described the detailed diversity of the cultural fungi from deep sea sediments. And this was done from the Central Indian Ocean from depths of around 5,000 meters plus, as a part of the deep sea project which was going on in the institute at then. And then the revolution came with regards to the next generation sequencing, what we call now, wherein a lot of people started, and then it became much easier to get the DNA and then start describing the unculturable fraction of bacteria as well as the fungi. ATNase and ITS are one of the most used primers for reporting this. And then people started exploring the different avenues in the deep sea. And then fungi from the hydrothermal vents were first time described in 2009 by Calves et al., wherein they not only reported the cultured but the uncultured diversity using the 454 pyrosequencing. And yeah, they could manage many more types of, to describe many more types of fungi than you could do in a cultured studies. This is to just give a brief idea about the locations which have been sampled for isolating fungi and describing the diversity. If you see, this is the area which I am going to talk upon, the Indian Ocean. There are not many studies or reports which are available. Obviously, there are logical reasons for that. It's not very easy to go and do sampling in the deeper oceans with the depths into picture and the logistical issues with regards to reaching that position. But even then, it is really underexplored, and we do not have much information when you compare to the Pacific and Atlantic oceans. And then this is like this report is giving about culture independent, the water column, the sediment, and the sediments below seafloor. That's the this I will talk to in my next slides, where I will be describing what the sampling techniques to study these organisms. And this is to just give you a brief about the different kinds of fungal genera and the morphologies which have been described from all the studies which are reported in the previous slide. So you see a lot of terrestrial cultures have been reported, like typical of Aspergillus, Acrimonium, and so on and so forth. Many of these non-sporulating forms and unicellular yeast have also been reported from. But then again, Indian Ocean is really underexplored as compared to the other oceans. With all these studies, there is a contribution of around 444 species. Number might be changing, but this is from a report which was published 3 years back. 444 species which have been described, including 7 genera and 10 species of Basidiomycetes and 177 genera from the Ascomycetes. A lot of information with regards to Indian Ocean has come from our group. We have been working like past more than 30 years now in this. Since 1992, Dr. Rohit Kumar started this work, and I have carried forward with now, and my students are doing it further. So this year we managed to describe around 221 different isolates got from different depths of Indian Ocean. And then we further improvised with with keeping the pace in the research which was there. We also adopted the culture independent approach, and we could see that we were able to describe quite a more number of species as compared to only the culturing aspects. And not only from the deeper parts of Indian Ocean, one of my colleague and Dr. Rohit Kumar's student, Dr. Catherine, managed to work upon the fungi which were present in the anoxic zones of the Arabian Sea. Like, let me just tell what is anoxic zone, because moment you say water and sea and oceans, people always have that feeling that, okay, there is enough oxygen because there are fishes living into the waters which are there in the seas. But that is not the case. Arabian Sea and Bay of Bengal are typical basins wherein just after the monsoon, you have a typical zone of around 500 to 700 meters of water column, which is devoid of any oxygen. And contrary to the definition which we have studied in our school times and in colleges and graduation studies, like fungi are aerobic. That notion was really killed by some of the discoveries which were done by mycologists now, where they have managed to obtain some of these fungal cultures from complete anoxic environments. And that's not only from the sediments, but also from the water, which is devoid of any oxygen. This was one of the first reports with regards to a culturable Basidiomycetes isolated from the anoxic zones. And then this also led to looking into their role, the nitrate reduction, because that is what happens as they use the nitrate as a ultimate electron acceptor, and that is how they carry on their metabolic activities in this area which is devoid of any oxygen, which is normally used as a electron acceptor in the normal respiratory chains. Yeah, this is a recent review which we have published from the with regards to the fungal diversity from the deeper parts of the Indian Ocean. Again, I mean, the fungal diversity is really not much. It's really unexplored and underexplored topic which a lot of people can look into. Now, let me talk about what and how we are doing these isolations from the oceanic waters. So this is one of the basic requirement for collection of the sample. It's called as a Niskin sampler, which is attached to this kind of attachment called as a CTD rosette. And this is lowered from the ship or the platform where we are working. And it collects the water from the desired depths. Now we are talking about depths of 5,000 meters. That's like 5 kilometers below the surface of the water. And then it is brought to the surface, and then you can carry on with your regular work with regards to your isolation or the DNA culture isolations or the DNA isolations. So this is about the water sampling. Similarly, these are some of the equipments which are used for collecting the sediments. This one is called as a Usnil box scorer, where you have a box fitted into this apparatus here, and this is released from the ship. And then once it touches the bottom, it collects the sediments, and then you get it on top and then start working with it. This is another one wherein these are small tubes here, which once they hit the bottom, they get filled with the sediment. They penetrate into the sediment there. So this can be used only when the sediment is little bit softer and penetration is possible. This cannot be used at a hard substrates. So this gives around 50 to 60 centimeters of the top surface sediment of the sea floor. But then if you are interested in looking into the longer surfaces, so this is called as a gravity corer, wherein this is a weight which is attached on the top of the long tube, what you see here. And with the weight, it slowly goes into penetration. Again, this can be used only when the sediments are soft enough. For that, you need to do a lot of geological surveys to ascertain the hardness or the softness of the sediments, and then only you can carry out. With the help of this kind of work, in 2003, we have published a paper from the Chagos Trench, wherein we could get a fungi, which we reported to be 0.42 million years old. That's published in Deep Sea Research. And this is, I think, at that time, this was the oldest known fungal records which could be done. And then after that, then you have this International Ocean Drilling Program, where they have this kind of drills. If many of you have seen that how they do the borewell digging in the terrestrial land for getting the waters. So this is also something similar, wherein they have a drill and they use it, and the course of length more than 2,000 meters have been recovered under the International Ocean Drilling Programs, which are then cut into, and then researchers look into, and then they have access. And this is where now people have recorded presence of fungi even from depths of 1,700 meters. Of course, using the DNA signatures, not the culturable ones. So this is what I showed you. This is what we have used extensively for our work in the Indian Ocean, wherein this is a sediment, the subsurface sediment, which gets filled in this box. It is brought on the deck of the ship, and then you have this kind of tubes inserted into it. The very interesting fact in this from this region is that every 1 centimeter of the sediment, it takes around 1,000 years to get deposited. So you can imagine this 50 centimeter box, from top to here, there's a variation of 50,000 years. This is approximations based on the sedimentation rates of in that region of, and then, so this at different centimeter levels is fractionated in the laboratory, and that is how we do the culturing or the DNA extractions from this. The methods of isolations we have used are the simple dilution plating, particle plating. Then along with that, we have developed in our laboratory a modified enrichment technique by incubating these sediments under elevated hydrostatic pressure and the low temperatures. Elevated hydrostatic pressure is that, like, if you have this water column, at every 10 meters, you get an increase of 1 bar or 1 atmosphere of the pressure. Like the room where we are sitting and I'm talking, you are listening, is having a pressure of 1 bar. So at 5,000 meters from where the sediments have come, the pressure approximately is around 500 times more than what we are experiencing. And even at that pressure, microorganisms, not only fungi, bacteria, and other some organisms are known to survive. So they must be really hardy to survive these conditions there. So this is the equipment what I was talking about, the high pressure culture vessel. And this cylinder is having the samples which we add along with the nutrients and then leave them for 60 to 90 days because at the high pressures, the metabolic activities are extremely slow. Now, there are some reports wherein they have showed the duplication time of E. coli, which is normally like in the best conditions 20 minutes. They have recorded up to 16 to 17 hours also. So you can imagine the slowness of the metabolic activities. So we have done isolations from 90 to 180 days also to get the cultures in the culturable form in the laboratory. But one drawback with all this sediment work is that we do not get any true culture loving, true barophile, because what happens is that by the time this sample it comes up from 5,000 meters to say surface water, the pressure alterations occur, and by the time it reaches here, it is at the normal atmospheric pressure. So the true barophiles may not be surviving. So most of the organisms what we have reported and what we have got in our laboratories are the ones which are able to tolerate the high pressure. So we call them at piezo tolerant. That's the barotolerant or pressure tolerant. But then there are some reports which not with regards to fungi, but for bacteria, people have done isolations using the sampling using the unmanned deep sea vessel. Like at present, the Japanese and French and Americans in Scripps Institute of Oceanography and Woodsole Institute of Oceanography, they have the submersibles wherein they can go and collect the samples and get them in that particular pressure, the in situ pressure, and then get them in the lab. We unfortunately, India still doesn't have, but then in the recent meetings and all, you might be hearing about similar to Samudrayan, Chandrayaan, they are doing Samudrayan, which is being at present designed by National Institute of Ocean Technology in Chennai, wherein they aim to devise a mechanism to do all this sampling in the deeper parts of the oceans and explore the unexplored. Coming back to what we got from all this culturing was, surprisingly, most of the terrestrial fungi were isolated from the sediments, be it Aspergillus, Penicillium, many of them are unidentified, Cladosporium, Curvularia. So there was nothing interesting or novel about the cultures what we got from this deeper parts of the oceans. So this is to just give an idea about what kind of different cultures we got from. This is the surface and this is the 50 centimeters subsurface depths. Many of them were unidentified sporulating fungi, which we were not able to identify using the available keys for identification. Mind you, this was all with regards to the culturable work I'm talking about, not the environmental DNA or the uncultured work with the next generation sequencing. But then it is very difficult to see these kind of forms in the sediments, what you get, because this is normally what you see under the microscope. And this is where we developed a technique for detection of this fungal hyphae directly under the microscope using a special stain called as calcul-4 or optical brightener, which is normally used to stain the chitin, which is in the cell wall. And that is where we really hit a jackpot, and we could see and record a lot of fungal hyphae present in the sediment, which were normally going unnoticed. And this could be one of the reasons where people are not really accepting the presence of fungi in the deeper parts of the oceans or in the deep sea sediments. Sometime back I said like most of the fungi we reported were Aspergilli and Penicillium and this kind of terrestrial forms. So it was very, very difficult for us to really convince the community that indeed this fungi are from the deeper parts of the oceans, because one of the drawbacks, as I told you, as the sample gets collected and the box travels from 5,000 meters to surface through the seawater, and that is a time there's a lot of contamination which might be happening with the spores or mycelia which are present in the upper depths of the water. So this was a question which was raised by the community when we reported first time this fungi, so many of terrestrial fungi from deeper parts of the oceans. Are they really from ocean, or they are the contaminants which have contaminated the sample on their travel from deeper parts to the surface? So this is where we started thinking about, and we then got into this. We developed some antibodies which were raised against the most commonly form which we got, Aspergillus terreus. And then, yeah, we went back to the sediments from where these forms are isolated and reported. And we could see that it was indeed having a positive presence in the sediment, and it was not a contaminant. And this really hit upon the nail, and we could publish our findings with regards to the presence of fungi from the deeper parts of the sediments. So we put forth a theory that these fungi which are there are could be originating from there, or these are the forms in the spores or mycelia which are getting transported with the wind or the terrestrial runoff, which ultimately die or slowly sink the mycelia, not the spores, because spores are really very hardy and they get clumped with the marine snow or similar organics, and they go and settle down, and they get buried into the sediments, which when we go for our, or anyone goes for their sampling expeditions, that is where you get them out and then give them favorable conditions to grow in the laboratory. And once they are adapted well, they start growing. And that is where we were very disappointed that many of these weird looking forms, which we first got direct observations from the sediments and the first isolations, ultimately turned out to be normal aspergilli when they were given like 3 to 4 subcultures in the laboratory. I mean, this was really interesting morphological adaptations which they showed. Like instead of producing a conidia, they started another conidia 4, and then so on. Then these are just few of the examples which I put there, but there were a lot of interesting this kind of morphological adaptations, which could be because of the stress which they were undergoing, and as during the first isolations we got them. These are some of the yeast which we got from the elevated hydrostatic pressure cultivations in our laboratory. We also recorded some microconidiations in some of the isolates what we got. You have conidia, then you get the vegetative form, again conidia, vegetative form. So this was again a new thing which we saw. So these cultures might really having a good amount of adaptations to grow in this deeper parts of the oceans. It could be morphological, as we saw, it could be physiological adaptations. I'm not going to go in the details of these adaptations because that's a different topic altogether. But then after doing a lot of work, we have seen that in our laboratory that the proteins which are being produced by these organisms could be one of the reasons. A couple of my students have already completed the thesis in this, and we have reported a lot of unique proteins which and stress proteins which are produced by these cultures when subjected to the pressures. Coming to some of the recent work which we are doing, and obviously it's the fungal reporting will be going on. When Dr. Shenoy was introducing me, he said about a sailing of 90 days in the Indian Ocean. So this is a project at present which we are working upon, where we are trying to construct the genomic and proteomic map of the biota in this whole region. And we already had one long cruise of 90 days starting from East Coast, going from the Indonesian waters to touching the Australian waters and coming back to. So here we have collected around 700 samples of sediment and water, wherein we are looking at different microorganisms, bacteria, fungi, and along with that some planktonic forms also. This was well covered by media. And yeah, another couple of years time we should be able to contribute to that empty spaces of Indian Ocean with regards to the fungal presence. So these deep sea fungi are still really an unknown fraction of the marine environment, and I would like to end my talk appealing everybody that really future marine mycology should also focus on the commonly overlooked, unidentified or unclassified sequences in the deep sea fungal datasets. You can really use the datasets freely which are there in the NCBI, which aside from being great biotechnological potential, could also represent key fungal species for evaluation theories about the origin of marine fungi and solve the unanswered question once for all. That I would thank my guides, Dr. Raghu Kumar, Dr. S. Raghu Kumar, Dr. Chanlata Raghu Kumar, my students, and my colleague Catherine. So thank you, MycoAsia, for giving me opportunity to present our work here. This is a cumulative work which has been going on in our lab for last 30 years. Thank you.
Thank you, Dr. Samir. It was a very authoritative presentation on marine fungi and fungi from the Indian Ocean. Now the floor is not floor. This platform is open for question and answer session for Dr. Samir Damare. Please. Yes, Mr. Jackson.
Thank you, sir. It was a very wonderful talk, and it was very interesting for me also, sir. Sir, you have done a lot of work in this fungi that is in the ocean from around 2006. But throughout this time, we have also come to know that the ocean water, mostly the Indian Ocean, has been polluted. There is a rise in temperature of the ocean water. Have you noticed any change in fungal diversity throughout your study?
That's really an interesting question, because this is exactly what we are going to address in our, when I say the genome mapping, because we really do not have any baseline data to talk about the change in the community with regards to what are the differences in the fungal diversity with regards to the changes in the oceanic waters, be it regards to the pH or the temperature. So with regards to pH, I would say that it will really not affect fungal fungi so much, because as you know, the pH of the seawater is around 8.3. So it's towards the alkaline side, and I would say if it turns acidic, it will be more favorable to fungi. I mean, it may not happen because ultimately it's going to be a decimal change of the pH. With all these 8.3 might go down to just by 0.01 to 0.02. But in climate, yes, it might affect the fungal diversity as compared to the pH, but that's also not too much, because the fungi are really capable of growing and adapting in different temperatures. Like as I was talking during my talk, they are not only present in the deep sea, because deeper parts, when you talk about, it's at 4 degrees most of the time. And the fungal presence has been also detected from the hydrothermal vent regions, where you have temperatures ranging to 200 to 300 degrees. So I would say, I mean, from whatever we have understood, it will really not affect the fungal community to that extent as it might affect the calciferous forms like the sponges and the corals.
Thank you, sir. If you allow me one more, can I ask one more?
Yes, sure, please.
Sir, I'm more interested in biodegradation. So recently I have read papers where they have found that mostly the fungal species from the ocean have the capability to degrade certain pollutants. Recently, this year also, University of Western Australia, they published a paper regarding that. So, sir, do you think that the Indian Ocean holds certain fungi that can biodegrade certain type of plastics? Can we do work on it?
Yes, definitely. This is really interesting, and a lot of people have started working towards it, because you have unexplored. This is what I ended my talk upon. You have a lot of unexplored fungal population present in the deeper parts of the oceans, which once you get in the culture form, you should be able to use for a lot of biotechnological applications, be it enzyme or the biomolecules or the drugs, secondary metabolites which are produced by the fungi. And fungi, as you know, need a substrate to grow upon. So they are the first ones to colonize along with the biofilm which is produced on the plastics or the microplastics, and it would be, they would really help in degradation, I would say.
Thank you very much, sir. Thank you.
Any more questions to Dr. Samir Damare? It's a good opportunity to get the answers from the expert itself. Yeah, Dr. Rajesh, please.
Dr. Samir, thank you for a wonderful lecture.
Thank you,
It was amazing.
to see your work expedition and representing India in the global platform. My question is about the Aspergillus species you're working around. Do they really come up in the normal format of sporulation, or because in slide culture techniques we are quite often find these microconidia formations, like conidia germinate form conidia in a restricted conditions. But I was wondering whether we have few Aspergillus TDS reported from Manipal University as infecting the eyes. So there also it was not sporulating, only forms some structures like globo structures or some pieces of mycelia. If from human eyes we got samples and we could culture and identify it. So I was wondering what kind of taxonomic like variations which we can expect in these kind of strains.
Yeah, thank you, Rajesh, for getting up this question. When we had this weird kind of sporulations being recorded from the forms we got from deep sea isolates, we started exploring more into this, and we saw that there are reports with regards to this kind of weird sporulations from the aspergilli which are put under any kind of stress. So this is one of the common features which aspergilli are known to exhibit, where the sporulation patterns and the kind of spores they produce is very different. But then, as I was saying, if you provide them with right or exact the the ideal nutrients for them, at least in our case for the deep sea forms, we found that the culture started growing to the typical aspergillus which we know of. So yeah, in the initial isolations, like say first 3 to 4 subcultures, we had this kind of different sporulation patterns and germination also. But then later on, they really stopped doing that. And that is where we actually lost many of the cultures. So maybe that might be happening in your case also. Initially, you might be seeing that. But once you really get them in the culture form to grow properly on the normal media, like say Zobel or malt extract, you might see the normal sporulations. You can try that.
Thank you, Dr. Samir. Can I ask one more added question in that?
Please.
Yeah. Because International Subcommission on Aspergillus, Penicillium and Aspergillus suggest malt extract as optional. So what's your take on this?
Yeah, malt extract is the one wherein we really use it to grow and then describe. But when you're going for initial isolations, I mean, we have seen that malt extract is really not good, at least for our cultures. Maybe because in marine origin, I would talk with regards to that, because the nutrient conditions are really not rich. They are oligotrophic, means you do not have enough nutrients. And actually these kind of media like malt extract or the rich nutrient media act as a shock. So what we do is many a times we actually dilute the media and use 1:10 to 1:100 times. And that is where we have found good amount of cultures growing.
Damodar, can I?
Dr. Sharma ji, please.
In the past, the lignicolous marine fungi from mangroves and driftwood were not isolated. Mostly microscopic work, and people didn't try to isolate the fungi. And the sequencing was not there. But in the past 10 or 15 years, many labs have been sequencing. So we have a sequence data of known marine fungi supported by morphology. So when you do this deep sea fungi, particularly the environmental samples, and you get these unknown ascomycetes, in future we will be helping you, because we will be getting with the structures and the sequence data. Maybe you will end up hitting some of our fungi from the driftwood or the mangrove.
Yeah, Dr. Sharma, that would be really nice and interesting to look into, because there are not many people who can identify deep sea fungal forms because of this different kind of morphologies which they exhibit. So we can definitely do that.
Yeah.
Okay, thank you very much, ladies and gentlemen. Dr. Samir Damare is available on his email, samir@nio.org. I have already shared his email address in the chat box. He is also available on Facebook, on Twitter, also on Instagram, and also on WhatsApp if you have his number. Anyway, please feel free to contact him about any issues related to fungi or marine fungi, oceanography. Thank you, Dr. Samir Damare. It has been a wonderful experience having you here and listening to your expert talk on fungi of the Indian Ocean. Thank you.
Thank you.
Yeah. Now I hand over this platform to Professor Samantha Karunarathna. Sam, are you there?
Yeah, I'm here.
Yeah, please take over.
Okay, thank you, Dr. Shenoy. So our next presenter will be Dr. Gunjan Sharma. She will be talking about transcriptomics of plant pathogenic fungi. Let me introduce Dr. Gunjan Sharma. Dr. Gunjan Sharma is working as an assistant professor in the Department of Plant Biotechnology at Gujarat Biotechnology University. And she performed her PhD dissertation research at CSIR Institute of Microbial Technology, Chandnagar, India, and received her PhD degree from Jawaharlal Nehru University, India. She has 6 years of postdoctoral research experience in the areas of plant pathology and host pathogen interaction studies in India and Israel. She is also the production manager of MycoAsia. So over to you, Dr. Gunjan Sharma, to enlighten us on transcriptomics of plant pathogenic fungi.
Thank you, Samantha, for the lovely welcome. And hello, everybody. Good afternoon. So it's a pleasure to give a lecture on World Fungus Day. Actually, it's my first online lecture, also online, offline, first talk after being a faculty. And I would thank Shri Venkateshwara University and MycoAsia for giving me this opportunity. So let's begin. So in this talk, I will be giving you a brief introduction about the plant-microbe interaction and various omics approaches that we can use in plant pathology studies. And then I will give you a brief insights about my research work. So in today's talk, I will be talking about plant-microbe interactions and various omics approaches that we can apply in plant pathology with respect to plant pathogen interactions. And I will also be providing some brief insights of my research work and future research gaps that can be explored further. Next, please. You can go forward. So plant microbe interactions can be good and bad. We all know the benefits of a healthy interaction. It supports the growth of the plant. Next, please. There's more animation to it. So it provides the plant, no, back. So the plant provides shelter and nutrition to the microbes. And in return, the microbes provide nitrogen fixation, biofertilizers, plant growth promoting factors, and also some endophytes help in biocontrol. But when this interaction is detrimental, that is the pathogenic form, it causes disease and death of the plants. So my talk is mainly about these pathogenic interactions. Next, please. So for a disease to happen, there are three different factors. There's an interplay of these three factors. So only one factor is not responsible. There has to be a conducive environment where the suitable pH, temperature, humidity for the pathogen is there. There should be a presence of a virulence pathogen along with the susceptible host. So only when these three factors come into play, the disease happens. Next, please. Next. So according to FAO, every year there is a loss of between 20 to 40% of global crop yield due to different plant pests. These could be fungi, these could be bacteria, these could be viruses. My work is mainly about fungal plant pathogens, and as reported by Dean et al. In 2012, this is the list of top 10 plant pathogens. Next, please. So every now and then we keep seeing different news reports about fungal diseases that are causing destruction to the overall crop yield and the quality of crop like wheat, different fruits, And fungal pathogens are usually the most common factors of most of these diseases. Next, please. And in this slide, I'm giving some pictorial representation of various plant diseases caused by different factors like fungi, bacteria, viruses, nematodes. Next, please. And as we all know, global food security is a very important issue, because if we don't take care of our plant health now, we won't be having adequate food supply in the next 20-30 years. So this is raising a global concern. And with this slide represents the top 4 fungal pathogens that are affecting various crops that are staple to more than 50% of the world population, such as wheat, plantain, rice, and soybean. Next, please. So how we can use omics in these plant pathogen interaction studies? We all know that DNA comprises of the genome, and when the DNA is transcribed to mRNA, it forms transcriptome, which gives an idea about the expressed genes in an organism. And when these mRNA are translated to protein, they form proteome. And further, from these proteins, different secondary metabolites are produced that forms a metabolome of an organism. My talk is mainly focused about the transcriptomics approach. Next, please. So using transcriptomics approach, we can use we can get system level understanding of host pathogen interaction. Next, please. So in the host side, using genomics, we can use we can get information about SNP details, QTLs, and candidate resistance genes. And also we can get understanding about different markers that can be used for future molecular breeding crops to generate biotic and abiotic resistant crops. And in the pathogen side, we can get a lot of information about different biochemical pathways that are involved in resistance and virulence of a host and virulence of the pathogen. And we can also get information about the different secretory proteins that act as candidate effectors that I'll be explaining in my further slides. And these are very important proteins that play a role in the virulence of a pathogen. And also using the comparative approach, we can gain information about the host specificity factors of different pathogens. Next, please. So now I'll be talking about my work, which I did on the powdery mildew of pea. So pea is a very important crop. You can go make all the animations here. Yeah, thank you. So this crop not only provides a lot of protein, which is a major dietary component of Indian population. And along with that, it's a vegetarian superfood. It provides fibers, proteins, vitamins, and minerals. Also, the dried plant parts, the pea straw, can be used as cattle feed. And the root of this plant improves soil fertility and also improves soil quality by increasing the organic matter, providing soil porosity, and also providing structure to the soil. So this is a very important crop. However, next slide. So this crop, there's more animation to it. Back, yeah. So let me show you how a pathogen infects a host. So here I'm taking example of powdery mildew disease, which is an obligate biotroph fungi. That means it survives inside the host tissue and without causing disease, and extracts nutrients from the host continuously for its survival. So as soon as the spore lands on the surface of the leaf or stem, wherever it's infecting, it provides enormous osmotic pressure and forms a penetration peg. And further, this penetration peg differentiates into a lobe-shaped structure known as hostorium, which is the primary site for the synthesis of these effective molecules. And also, this is the primary site for the uptake of nutrients and water molecules from the host. So this is the key structure that is providing nutrition to the pathogen, and also it's secreting effector molecules. So now what are these effector molecules? These are the small secreted proteins which are usually cysteine rich in nature. They are very small in size, less than 200 amino acids long. And these are the key players that have some role in suppressing the host defense mechanisms and infection process. So how we can gather information about that? So next, please. This is the real images of an isolated Hostoreum from powdery mildew infected pea leaf that I did myself in my work that I'll be telling further. So in reality, powdery mildew Hostoreum looks like this. Next, please. So powdery mildew, no, back, yeah. So powdery mildew is mainly characterized by white powdery appearance on the aerial parts of plant like leaves, shoots, and fruits. In case of pea, a whole pod is affected by powdery mildew and it leads to crop loss up to 40%, and also that affects the quality of the pea seeds. So since I already explained this is an obligate biotroph, that means it survives inside the host leaves and pods. So what I did, I isolated the Hostoreum from powdery mildew infected pea leaves using isopycnic centrifugation approach. And then I extracted RNA from this sample, from the isolated cells, and submitted this sample for transcriptome sequencing, for RNA sequencing, mRNA. And further, using a bioinformatic pipeline, next please, I identified 167 candidate effector proteins that fulfill all the criteria of being small in size, having high expression, and cysteine-rich motifs. And then further, next please. So this is the circles plot showing the expression value in the orange and yellow layer. The central cladogram represents the different clustering pattern of 116 proteins. So they were divided into 13 protein families. Most of them were unannotated based on interproscan, because that's the one of the characteristics of effector molecules, that they don't show homology with existing virulence genes. Every pathogen evolves on its own, and that's why they're very host specific in nature. And the outer layer represents their name, and the inner two layers represents the motif present in them and their similarity with some of the related powdery mildew genes. I would not go into that much detail, but next I would like to show you that I wanted to check their expression pattern. So based on this bar graph, which was generated using qRT-PCR experiment, where I had two different samples. One was isolated from Hostoria, enriched Hostoria, and one was just the surface mycelia on the leaf. And the black bar represents the Hostorial expression. And as you can see, all the effector candidates were highly expressed in the Hostorial sample, which is their characteristics. Next, please. Next, I checked their back. Yeah, next I wanted to check some candidates. I selected some of the candidates and performed a time course qRT-PCR experiment where I wanted to see their expression pattern in different stages of fungal infection, like formation of presoria, which is between 0 to 6 hours time point. The x-axis represents the time point, and the y-axis represents their expression. So as we can see, different candidates showed different stage-specific expression patterns. Some of them were highly expressed in the formation of episorial stage. Some of them were highly expressed in the late stage of infection, and some of them were continuously overexpressed. Next, please. Further, I selected two candidates from one of the family that was really coming a lot highly expressed, and that is one of the widely reported effector family in powdery mildews, which is known as RALF effectors. This is called as RNase-like protein associated with Hostoria, RALF. So these three candidates, EPCSEP1, 9, and 83, 1 and 9 belong to RALF effectors, and 83 was showing some similarity to another similar powdery mildew species. So I took 3 candidates, And I saw that when I try to silence this transiently in the pea leaves, upon silencing, next please, you all can see there is a visible difference in reduction of virulence symptoms in these silenced pea leaves. So left is the GFP silenced leaves and the right is the candidate effector that was silenced. So as you can see in the pea leaf itself, you can see visible reduction in the symptoms, which shows that these are the molecules that are indeed having some role in the infection process. Also, I quantified the microscopic features, and as you can see in the D panel, most of the spores were not able to germinate upon silencing. 1 minute. Sorry. Yeah, so I had a bit of cough and it just started, so I'm sorry for that. Next, please. I also performed some subcellular localization where I tagged these candidate proteins with GFP marker, and I wanted to see where it's localizing in the plant cell, the host cell. So as you can see in the left figure, candidate, sorry again. So the candidate 1 was localizing only to the nucleus. Candidate 9 was localizing to nucleus and the cytoplasm. And candidate 83 was localizing again to the nucleus. The last one is the empty vector, which is localizing to both the cell, cytoplasm, as well as nucleus as expected. And I also performed some homology modeling studies by comparing the protein sequence with already known Ralph effector candidates. And as expected, the RNA's active site was present in these two, candidate 1 and 9. So all these findings indicate that these candidates have some role in the virulence and they possess RNA's activity. Next, please. So this study was published in Molecular Plant Pathology Journal. You can go read this paper for further details if you're interested. It was also the cover page of the that issue. Next, please. So we learned, I gave you a brief of how we can use transcriptomics in plant and microbe interaction studies. And the research areas to be explored are enormous. We can find about host specificity factors. I'll be further dealing with those in my future work. We can get enough knowledge about differentially expressed genes by comparing different stages, resistant and susceptible cultivars, different pathogen infection stages, different pathogens together. So there is n number of possibilities that we can do using omics approaches in plant pathogen interaction. We can find pathways and mechanisms that are involved in virulence of a pathogen. We can identify effector candidates. There's the entire secretory protein prediction pathway that is part of this paper. And so you guys, if you're interested, you can look there in the methods, and the entire pipeline is very user-friendly, and using that, we can predict our effector candidates. And all this information will help us to improve our crop variety using the new age genome editing approaches. So with that, next please. I would like to conclude my presentation today, and I would like to thank my funding from CSIR, DST Serve, and ARO Israel for helping me progress in my career, and all my mentors from PhD, Dr. Belle, Dr. Anil, my postdoc mentors, Dr. Divya, Dr. Stanley, and my best mentor, Ms. Marcel, and all my colleagues and friends for helping me throughout my journey. So with that, I'm open for questions. Next slide. Thank you for your time. Let's celebrate fungi together. And you can reach me on my email ID if you have any questions. Thank you.
Okay, thank you very much, Dr. Gunjan, for a very insightful presentation. And now the platform is open for questions.
Thank you, Dr. Sharma, for your wonderful talk and sharing the transcriptomics actually. At the end of your slide, I saw that you mentioned about pathways. I'm just thinking, in case you have explored about the the differences among the in the comparative pathway level, let's say the pathogenic fungi in the host plant, and maybe with different kingdom, let's say parasites. In case you have explored, and if so, could you please pinpoint to some case studies?
Okay, so this one I haven't done. I haven't gone that far of comparing insect candidates versus insect pathogen with fungal pathogen yet. But yes, I'm thinking about comparing different pathogen types like biotroph versus necrotroph versus hemibiotrope. So this aspect I'm thinking already, and I'm writing some ideas, but I haven't thought about comparing insect pathogen.
I mean, since you are, doing this large-scale transcriptomic study, I guess it will be a wonderful sites to go on because it will be then more applied. Then you will dig into more applied part of it. And I'm just one. I thought maybe you were already in that.
Well, that's a wonderful suggestion. Thank you for that.
Yeah, thank you. Yeah.
Yes, please.
Dr. Rajesh?
Yeah. Dr. Gunjan, thank you for a wonderful presentation. It was indeed a delight to hear omics from India with the support of Israeli eminent scientists. My question is related to a pathogenic fungus, maybe Colitotrichum. I have read somewhere pre-suria production helps it to become more pathogenic rather than in some species it is not. Is it a case really we can prove through your kind of studies? And the other question is related to pathogen and endophytic differentiation in species like cosmopolitan species like Colitotrichum Cleosporus. Can we do it through omics studies?
Yeah. So regarding your first question, in Aprisorium structure, usually the melanin biosynthesis gene, they are very active. And they are like one of the known candidates that have been already shown to have role in virulence of Colitotrichum species. So that is one aspect to it. The second question that you asked was about that study has already been done. It was published in 2014 in Nature by O'Connell Group. And there they have compared Colitotrichum graminicola and Colitotrichum higginsinum. And they have compared the hemibiotrophic and the endophytic stages and using genomic and transcriptomic approaches. And they have done a detailed study about these lifestyle transitions. So please check that paper. That's a very good one. It's in Nature.
Thank you, Gunjan. Thank you for the clarifications.
Thanks.
May
Yeah.
Yeah. Am I audible?
Yes, yes.
Yeah. First of all, thanks for that informative lecture. I just want to know that you are just doing this kind of work in the in silico. Are you planning to do some in silico work or?
So I began with in silico, all the bioinformatics, the circus plot that I showed. That result came from in silico bioinformatic analysis. And then later in my slides, I showed some molecular biology experiments that I did. So always when we want to dig into the expression level, the gene expression level, we have to use the omics approaches. And of course, all the bioinformatics tools help us to understand our data better. So yeah, it started with that.
Yeah, because I'm just curious to know that all kind of these kind of work we have to ultimately have some sort of initial idea, as you have mentioned that through this bioinformatics analysis and in silico work, actually we had some substantive idea that how we can proceed to the wet lab experiment. So that's why. Thank you so much for that clarification. That
You're welcome. The information can also be derived from genomic sequences that are available in GenBank. But for more deeper understanding of what happens during an interaction, you have to go for transcriptome level. It will not come from genome.
okay, thank you so much.
Yeah, thank you, Dr. Gunjan. Can I ask the question now? This is Ramaya.
Yes, please. Hi.
Thank you for a wonderful and very lucid presentation. I'm not a mycologist, but I have a curiosity to know, now that you have knocked off the hosterium part of it using your molecular methods, are there natural analogs that naturally occur in some, crop fields of peas where you don't get this mildew disease, but then what is the cause, if you know? If you can extrapolate from your experiments that you have done, how are the natural analogs similar to the one that you use for knocking off the component that you don't want in the pathogen can be kind of understood? There can be many, I don't know, but please can you elaborate and educate? Thank you.
No, it's a very interesting question. I haven't thought in this area of comparing our biological molecules with the natural analogs in the field. But yeah, I don't know. I think I'm not prepared to answer this question at the moment.
No. I really didn't know to show you. I'm a generalist. I know you can ask Dr. Shenoy, your mentor or boss. But it was the wonderful talk. So I enjoyed. A thought came to my mind, thinking that now that this lady has done a very interesting experiment wherein she could prove that you can arrest the expression of hostorium-based exchange of the smaller molecules that you said are so important in its virulence enhancement. Thought that maybe in compared to the fields, in some places the mildew is taking a lot of toll on the crops, whereas in some not. Therefore, are there some kind of a mechanism that we understand the soil biochemistry so much so that the inhibitors naturally exist, and therefore the crops are successful,
So
or otherwise?
I can only think of one possible answer right now is to compare the soil microbiome of these two different locations, like one with a disease and one not with disease, and then see what difference in the secondary metabolites production is there in the microbes or microflora of those two kind of soils. So that's the only thing that comes to my mind right now.
Wonderful. I mean, if I may be allowed to ask this question, how about taking this work further and proving that this is an effective means of, curbing the hostorium expression so that the plants become successful? And your own, probably this is novel. I don't know if it is, and if it is so, you please explain. And that I find this is a kind of an insight into your transcriptomics based application for curing the diseases. Yeah.
Right. So regarding this, usually we have different protein-protein interaction methods. So let's say if we know what candidate is causing disease, so we can find the corresponding protein in the host that it is acting upon, and that it's suppressing some defense mechanism, some pathway. So we can find those candidates in the host, and then using genome editing approach, CRISPR-Cas9, which is very popular these days, and it has been allowed by the government, Indian government also, without the use of any foreign element. So using this, if we know the target in the host, we can easily knock it out and produce disease-resistant crops.
Thank you very much. I'm done. Thanks.
Thanks.
So can I ask one
Any
question?
more questions?
Yes, please.
Yeah. So Great work. So actually, I just wanted to know, like, when you do analyze food and when it's novel, like here you have one side, but you don't have a transcriptome available. So in that case, how do you actually do assess the folds in the expression level of certain genes?
Can you type your question, please? I can't hear your voice is breaking. Can you type your question in the chat box? And meanwhile, I answer it.
Sure. I will check. I'll try.
Yeah.
Ma'am, shall I? Ma'am,
Yes.
I'm a postgraduate student and I don't have a deep knowledge in it, but I just asked a basic question that I have heard about the mycoviruses that are present in these pathogenic funguses. So, can you share anything about that? Can we use them as biocontrol agents and something like that?
What word you said? Micro?
Mycoviruses, the viruses that infect the
Mycoviruses.
funguses.
So I haven't studied mycoviruses. Sorry, beyond my scope right now.
Like we have bacteriophages that infect bacteria. So can
Yeah,
we
so there's
utilize that?
mycoviruses that infect fungi, but how they can be a biocontrol agent? Sorry, I can't answer this now. But I'll get back to you.
Okay, ma'am. And the talk was very impressive. So I thought about like implementing those techniques and all to somehow move this topic further. So
Sure. There are plenty of research articles available now. Everybody's using transcriptomics. And there's a term called simultaneous transcriptomics. There is a term called comparative transcriptomics. Just Google these and you'll find good articles. Good luck.
Thank you so much.
Gunjan, you have a question in the chat box.
I think to assess the fold change, you need housekeeping genes and reference transcriptome sequence. Like if you will design primers, you'll have some sequence, right? It will come from somewhere. It will either come from homology based from some related fungi or from the transcriptome or the genome itself. So in the similar fashion, we will do the designing of primers for the housekeeping genes, and that's how we assess the fold change. The reference transcriptome might not be available, but there is another approach called, there is one mapping technique, and then there is there is reference guided mapping, and then there is de novo mapping. So you can use that. It doesn't need a reference genome. It aligns based on the contig length and alignments. So using that, you can have your own transcriptome sequence of longer sizes, and then that way you can annotate and find the gene of interest and also the housekeeping genes.
Any more questions from the audience?
Good evening, ma'am.
Hello.
Hello. Good evening, ma'am.
Hi.
Ma'am, I want to ask you one question.
Yes,
regarding what is the basic difference between endophytic fungi and pathogenic fungi?
Easy answer. Endophyte don't cause disease. But yeah, if you go to molecular, again, you have to compare the genome, their key molecules, what's the difference. That comes under the area of comparative genomics. But endophytes are usually surviving inside the host cells without causing any visible disease symptoms, and it just derives nutrition and it's beneficial. Most of the time, it's a symbiotic relationship. for endophytic fungi. The pathogenic, of course, it survives inside the host and also kills the host cells.
In genomic level, how to find out it?
So for that, you need to go for the comparative genomics approach, where you will be comparing different molecules, different molecular signatures, carbohydrate, degrading enzymes, secondary metabolites, different transcription factors, and then you will be assessing what's different in what enriched in what. You will be going for gene ontology study and all kind of different bioinformatic analysis, and then you can compare the two genomes of one of your reference endophyte and one of your pathogen. It can be from the same genus. It can be from the two strains of the same fungal species. One could be behaving like a endophyte, one could be behaving like a pathogen. So for that, you need genomics approach. Also transcriptomic if you go to for the expression level.
Thank you, ma'am.
So if there are no more questions, we can move on to the next talk. So thanks again, Dr. Gunjan, for enlightening us on transcriptomics. It was a wonderful talk. Thank you very much.
Thank you, everybody. Thank you.
So we are moving to the last talk. So the talk will be done by Mr. Parimal Udgave. So let me introduce Mr. Parimal. So Mr. Parimal studied Master of Science in Biotechnology in the UK, and he is the founder and director of BioBrittle India. And his current research areas are medicinal mushrooms, especially cordyceps and ganoderma. So he'll be talking about healing with mushrooms, medicines of future. So over to you, Mr. Parimal, to enlighten us on medicinal mushrooms.
Thank you for introducing me to the audience, and thank you organizers for giving this opportunity to me. So first of all, good evening to everyone. So basically, we will be talking about the medicinal mushrooms, where we can use this in different applications, especially in medicines or pharmaceutical industry, basically. So basically, we will start with the history. So basically, medicinal mushrooms have an established history of use in traditional ancient therapies, especially in Chinese medicines. Other than that, contemporary research has validated and documented much of this ancient knowledge, especially about the ganoderma mushrooms in Chinese traditional knowledge, medicinal knowledge. The interdisciplinary field of science, which studies medicinal mushrooms, has been developed, and for the last 3 decades, it continues to demonstrate potent and unique properties of compounds extracted from a range of mushroom species, especially simply mushrooms are fungus, so for fungus species. Modern clinical practices in Japan, China, Korea, Russia, the USA and France, and several other countries rely on mushroom-derived preparations. So basically, we are not seeing one second. We are not seeing more transitions in India regarding medicinal mushrooms, but everywhere except India, actually, more mushrooms, more medicinal mushrooms and products are coming into the market. Basically, in the China, especially the Lingxi or Ganoderma mushrooms and shiitake mushrooms are more popular, and ancient knowledges, they are discussed more, and their properties for human health, human medicine, as a human medicine, basically. So you can see Ganoderma mushroom here. So simply fungus type, one of fungus, a popular medicinal type right now. And then the shiitake mushroom is there in the second photo. Basically, mushrooms have a lot of species, but we will be focusing on much more which are related to medicinal applications. mushrooms have played very important role in different countries, especially in Eastern European countries. They are used to treat the ailments where they are used by rural populations. In the other important species, chaga, then woodconch mushroom, then birch polypore, and tinder bracket. These are some popular mushrooms you may have heard in the market or in the studies. These mushrooms specially used for gastrointestinal disorders for various forms of cancers, bronchial asthma, night sweats, etc. Here you can also get, you can see different pictures right now again for the chaga, woodconch, birch polypore. There is also a long history of traditional use of mushrooms in different Mesoamerica, especially psilocybin in Africa and Egypt. A very special role was found in Amanita muscaria mushroom, which is the photograph is given here, in Siberia and Tibetan shamanism, Buddhism, and Celtic myths. So basically, this is how the mushroom looks like, the psilocybin and Amanita muscaria. So in general talks, actually, mushrooms or fungi simply are extremely abundant and diverse worldwide. So nowadays, medicinal mushrooms are commonly used for different applications, were categorized as dietary food, dietary supplements, then natural biocontrol agents in plant protection, or even related to antibacterial activities, especially. Then the cosmetics, basically. Then in the new class as a mushroom pharmaceuticals. So these are the way how mushrooms can be categorized in our applications. There is one more point where mycotherapy is referred. So basically, healing with mushrooms or mycotherapy is ancient discipline with 5,000 years of history, but it is not well known all over the world. In recent decades, it is legitimized by science, which has recognized its enormous potential. Indeed, mushrooms can maintain body's equilibrium, health, and when there is some problem or disease, restore well-being. So basically, mycotherapy is a new science which is coming, especially in India, I would say. So in the in the recent years, it will develop as well. So it studies and uses mushrooms to improve human and animal health. So these are some examples why mushrooms or fungus, edible fungus, are better than herbs. So mushrooms give more effective results than herbs and plants. Here some are examples, such as in inflammation and allergies, Ganoderma is better than Ribos nigra. Like circulatory disorders, auricularia mushroom is better than Ginkgo biloba. Then for gastritis, hericium, then papaya plant, basically. In prostate diseases, Gryphola mushrooms are better than Serenoa repens. In flu diseases, Agaricus is better than Echinacea. In hepatitis, Cordyceps is better than Taxacum. And in kidney failure, Polyporus is better than Equisetum. So these are some examples where mushrooms are proven to be better than herbs as compared to different plants or plant compounds or plants or herbs, basically. So when we come to the point, mushroom is basket of invaluable substances. So especially rich in beta glucans and terpenes, which is actually secondary metabolite. And the next thing is like vitamins, lot of vitamins such as vitamin B, C, D, where mushrooms has good value. Then in minerals, proteins, antioxidants, and basically they restore or strengthen the body's state of equilibrium. So we are not really proving that the mushrooms can heal your diseases, but technically they are maintaining, restore and strengthen your body's state of equilibrium. So basically you can defend with any diseases or health conditions. These are some top mushrooms, top medicinal mushrooms we can talk about normally. So first is like oyster mushrooms. So basically, the active constituents of these mushrooms, you can see here beta-glucan, polysaccharides, antioxidants, amino acids, minerals, vitamins, and protein. Basically, oyster mushroom is known for its protein content. There are some uses here or effectivity is given where they are protein rich, immune system booster, heart health, fourth is like antibacterial activities, and fifth is weight loss. So here are some basic protein source of mushrooms. So simply oyster mushroom is getting popular worldwide for the protein rich content, and the second thing is like as a normal immunity booster supplement. Other than that, the second is like Ganoderma mushroom, also called as linseed in some places, and reishi as well. Reishi mushroom is also named for Ganoderma mushroom. Here, active constituents are basically most important is polysaccharide, where they have triterpenoids, adenosine, same as proteins, amino acids, vitamins, lot of constituents actually here. But here you can see one thing, which is like they have secondary metabolites as well here. Ganoderma is more effective in Alzheimer's disease, prostatic hyperplasia, BPH, cancer-related tiredness, diabetes, hepatitis B, cold sores, asthma, and bronchitis, and then the cancer as well. So I have seen more demand right now coming in India, especially for the Ganoderma mushrooms, where they are commonly used in diabetes, especially. And the second thing is the immunity booster products. Actually, the third thing is like shiitake mushroom. Simply, they are more popular in Japan and China, but they are also coming right now more in India as well. So basically, active constituents are polysaccharides, triterpenoids, geranium, adenosine, proteins, amino acids, and vitamins. They are also effective against cancer, immunity booster, kidney health, heart health, cancer-related illness, antioxidants. Then the next top mushroom right now, I would say in terms of the market and the pricing is cordyceps mushroom, because this is completely artificially grown in the lab, in the laboratories, basically. And their active constituents are mainly two, which is cordycepin and adenosine. Basically, these mushrooms directly gives ADP to your body, which can help your body to regenerate or generate the ATP, which is responsible for your energy, or also to the stamina, or simply to the moods as well. So basically, it is effective against cancer. It is also common immunity booster, kidney health, heart health, and cancer-related illnesses, and antioxidant. But it is more commonly right now used in beverages, where you can use it in coffee or teas. Then some products which are mixed with other mushrooms as well. These are some modern day products. Actually, we are doing some work on this. So these are like 9 mushrooms, where some mushrooms are responsible for your skin, some for the cancer illnesses, some for energy, some for your brain health, and a lot of things like for your heart health. So every mushroom is responsible technically for single purposes. For example, oyster mushroom is commonly used for proteins, cordyceps is for energy, Ganoderma is for diabetes. So simply, this is our modern day, or simply we can see this is our future, where we can grow these mushrooms in lot of scale actually, and we can start utilizing it in different pharmaceutical applications, such as even the cordyceps is being used in some cancer chemotherapies, and different products are different applications, different possibilities are there. These are our some this can be your product, this can be your modern day product, where you can find dry mushrooms, mushrooms in the dry form, mushroom powder, extracts, capsules, and mushroom tincture. So basically, in India right now, we feel that the transition is coming where we will be using more mushrooms in some near future. And this is just initial stages in India. So hopefully in 1 or 2 years, mushrooms will be known by everyone in India, where they can use for common diseases such as diabetes, malnutrition. You will be using this. So basically, this is about the mushroom, where we can use in medical, clinical applications in our human health researchers. Thank you for organizers. We'll be able to answer your questions if you have.
Yeah, thank you very much, Mr. Parimal, for enriching the audience with medicinal mushrooms. So now the platform is open for questions.
Unveil mushrooms, please.
Yeah, hello. So I have a question related to the economics of medicinal mushrooms, because cordyceps, like sometimes I'll show that 1,20,000 rupees in some newspaper article, and then they are sold at 3,000 rupees for like a kg. So that is a huge difference. So we don't know at what economic pricing is the product made, first of all. Secondly, on my last visit to Dubai, I visited many chemical stores, chemist stores, medicinal stores, where I found out that reishi mushroom, which is very much applicable for cancer, was not available there. People were, I asked, my whole research was that based only. They were like, mushrooms are not giving us better results, they're slow results. So pharmaceutical industries are giving up on mushrooms because of, one, the price factor, second, because of the trust, because like 3,000 and 1,20,000 is a huge amount gap. So there is lot of mistrust that there is uh, like addition of dirt or any other product with the cordyceps powder. So yeah, these factors are actually creating a misgap and uneducating the public, which we are actually trying to educate so difficultly.
Yeah. Your question is right, because actually I would say you're right about the pricing, but there are some very important aspects in terms of biotechnology and microbiology. So I guess you are you might be growing these mushrooms, or you might be growing different mushrooms, but for selling mushrooms, really, you have to be a pharmacist, I would say simply, in the pharma sector. Because when we are giving normal talks on mushrooms, like they will be used on cancers and other points, if you really, as a I mean, I have talked with a lot of companies, including bigger names in the India as well and other sites as well. The main difference about the pricing is because first of all, in India, when you are growing mushroom, it should be more technical, first of all, more scientific. Especially like mushroom, like cordyceps, you have to be a simply microbiologist, biotechnologist. So when you see a lot of people are giving trainings on cordyceps basically, but more people are also failing into cultivation because they are just thinking about money. They are not really searching about real uses of these mushrooms. That's what I think. Because when we are selling this to market, according to me, the mushrooms should be at cheaper pricing. First of all, as long as they are making you profits, you can sell the cordyceps like 50,000. In our economics, actually, 7,000 is our price for input for cordyceps. But as long as you are selling it at 50,000, still it is good margin for you basically. So if you're really thinking that mushroom can be sold like 1 lakh, 2 lakh, that's really it can be, but in more pure form, such as cordycepin or active ingredients can be isolated and then it can be sold for more pricing in the market. So I would suggest that instead of doing or going everyone for the selling, you have to concentrate on the applications first of all, and then you would be able to find actual buyers for this. That's how we work actually. And for doing this, you have to go for some literature. That's what I would recommend, where the applicability or applications can be seen actually.
Yeah, thank you. But with Rishi, that was with cordyceps. So what would be the cordyceps in level for around 50,000 rupee uh, cordyceps pricing
Yeah,
according to
yeah.
you?
Actually, the basic parameter is like at least 8%. We have seen in the different industries, which is 8 to minimum 8% should be there actually. So this is the pricing for the pricing I have given is like for 8 to 10%, you can say, not really maximum than that actually, because the
Yeah,
branding and
they
marketing because branding and marketing plays very important role, where you want to connect with the connect with the buyers. You have to be very professional. You should be looking very scientific as well to them. Then they will, they will try to buy from you. That's what we have experienced during visits and meetings with some people.
Okay. Now coming to reishi mushrooms. So reishi mushrooms, I am a user as well as a producer because I have gone through two major surgeries in a year, brain tumor. My mother is a cancer patient right now. So I've seen effects, but slow effects. I've educated my clients, I've educated retail clients as well as B2B, uh, like you said, pharma companies. But still, it is very hard because they come to the point that they say that we don't trust proper AC powder or extract because what kind of extract you give, like ethanol or water soluble. So, like, there are many constraints which are leading to, I think so. What my point is that people like you, me, and other like mycologists as well as mushroom farmers should get together and make a committee against, not against, for like the development of these mushrooms.
Yeah, I agree with you. But if you see in the industry, we are missing very important point, which is isolation and extraction. So basically, I would say in India, a lot of people are growing mushrooms, but they don't have any facilities, or they are not really trying into going deep into the compound level. So that's where we lack the market. That's what I always think, that we have to process these mushrooms in extracts very more scientifically and more deeper level. Then we would be able to show more results to pharma industry as well, and they will also trust more to us actually. That's very important right now.
Okay, fine.
Thanks.
We can agree on that. But I would seriously like to put this forward that people with medicinal mushroom knowledge should come together as a group and really educate the audience, the clients, as well as even producers. So even they should know that, like this is the way it should be made and there is no mistreatment with the efforts that we put in.
Sure.
Yeah. So thank you. It was a great event. Dr. San, even like Dr. Shenoy, you are my like the best thing that ever happened. And thanks a lot.
Dr. Rajesh, thank you. Thank you. Thank you. Dr. Rajesh, do you have any question?
Yeah, Parimal, I have a question.
Yeah,
you must be following Paul Stamets, isn't it?
Yeah.
So my question is around, like, other than cordycepin and psilocybin, do we have data, clinical trial data for chaga, reishi, and other, bracket fungi from across the globe? If it is so, is it in pure forms or in a crude form?
Actually, according to me, whatever I researched or seen the papers, most of the time they are in the crude form actually. So basically, crude form really doesn't apply in very effective way. Basically, that's what I think actually, because there are some more compounds in the powders, for example. So according to me, actually, very few researchers are on the clinical trials, but still they are very bigger clue right now in mushrooms, especially because hopefully this will get more clear when more people research or do clinical trials. That's what I would say. Like few researchers, but still big clue for us to go for that actually.
Anyone specifically from India, Oresh?
I would say in India, very less or very low. I mean, I would say at very smaller proportions, it is growing. But if you see in USA, Finland, for example, or different countries, especially USA, you can find face creams basically. You can find some cosmetics which are based or derived from mushrooms. But in India, it will be very hard for us right now at this moment because the big pharma players in India are not really right now convinced about this. But I would say some middle, I would say some smaller companies, they are trying for this because we supply to some companies in India, basically based in South India actually, and they are doing a lot of work on the cordyceps at a clinical level because especially they are directly exporting to some places in Europe. But I would say in India, it is not really more faster, but it is slow right now. But anyway, we are heading towards that actually.
Thank you, Parimal. Appreciate your reply.
Can I? May I? So, Dr. Rajesh, chaga is actually made or found more on the birch tree. So, it's very difficult to cultivate it. And so it's a natural or a forage mushroom. And its benefits are more. Lion's mane is one of the mushrooms that we can work on as a medicinal mushroom, which is also very, very effective on the body as well as equilibrium of your body, overall health. So that these three mushrooms right now are the main medicinal mushrooms in India. Thank you.
Yeah, yes.
Thank you, sir, for clarifications.
So there is a question on the chat box. So Jeevana Lakshmi, she asks whether there are any reports of usage of the mushrooms in Ayurvedic medicine.
Actually, I would say not really in India. We don't have really more deep reports or clues where it is used in Ayurveda. But actually, especially Chinese medicines, it is more written and more given more reference or citation as compared to India.
Yeah, this is
You
very
can't find
interesting. I also checked in Sri Lankan traditional medicine and also Thai traditional medicine. There's almost no reports regarding mushrooms in medicine. So it's very interesting. Are there more questions? If not, Dr. Shenoy, over to you.
Thank you, Dr. Sam. Thank you, Professor Sam, for conducting these last talks. Thank you very much. Now we are moving to the valedictory session. I invite Professor Paramageetham onto the platform. Okay, Please switch on your video. I invite Professor Samantha. I invite Professor Rajesh Kumar to the main portion of the our platform. Please switch on your video, Dr. Rajesh, Dr. Rajesh, are you there? Might have left. Anyway, but we would like to know the feedback from the audience. Maybe one or two people can give their feedback about this today's event. Please, you can raise your hand and talk. Yes, please.
Sir, really, all this presentation, it was very, very, very informative. And I am from Tanzania. My specialization is on lichens. So I had a very, I was very interested to ask question. And really, presentation was very good. And if you have any more presentation or any more session, please mail to us so that our students may also be participating. Thank you so much.
Thank you, Dr. Kalidas. Thank you very much for your kind words and kind feedback. Thank you. Any more feedback about the event from the audience, please?
May
Mr. Jackson, please.
Sir, it was a very informative session. It was long, but I really learned a lot. And my interest was in wood decay fungi and something about the deep sea fungi and mostly on species versus species complex. And every talk was very interesting, mind-blowing, and it somehow inspired me to do more into the research line. Thank you very much.
Thank you, Mr. Jackson, for your feedback and your kind words. Your words mean a lot to us. We'll work harder, we'll work better, work smarter in coming events. Thank you. Any more feedback about this event, please? Maybe the last, Ms. Shah.
Good evening, sir. I am pursuing my PhD and my topic is mushrooms. So it was really helpful for me and all the sessions were very wonderful. Thank you, sir. Please let us know for further session if it is. Thank you, sir.
Thank you, Ms. Shah. Thank you very much. We will let you know. We have a Facebook group, MycoAsia online training programs, where we share all our updates there. Please do subscribe to that group. Okay, thank you.
Sure, sir. Thank you.
Ms. Jeevana.
Good evening. It's really wonderful that I have been listening to all the lectures right from the morning, and they were very informative. And I think students who have participated have been exposed to variety of topics in mycology, and they're going to derive a lot from this lecture. Thank you for organizing committee for having such a wonderful presentation. Thank you.
Thank you, Ms. Jeevana. So, Ms. Namita Singh, please.
Yes. Good afternoon, everybody. This is Professor Namita Singh from Guru Chambeshwar University and the General Secretary of Association of Microbiologists of India. Sir, I just joined this group because some of my friend is from the physics department. He just transferred to me that it might be helpful for me, and I joined it. Really, it's a wonderful, you can say, the presentation and all. And I love to invite a few of the speakers in the forthcoming association meeting at Rohtak, MDU Rohtak. If anyone from the group want to come to join our international conference going to be held in the month of February, please contact me on my email address. My email address gs2020ami@gmail.com. So it was very wonderful, good presentations and all. I really appreciate the efforts of organizers. Thank you very much.
Thank you, Professor Namita Singh. I
can share my email address here, and I invite you all for the forthcoming conference. Yes.
Thank you, Professor Namita Singh. I was in Mysore. I attended AMI. Yes. Thank you very much for your kind words. It means a lot to us. And definitely some of our speakers, some of the people, experts from our MycoAsia network will be happy to, contribute to your event in February.
Okay.
Thank you so much. And it means a lot to us. Thank you.
Okay, thank
you. Mr. Shailesh?
Good evening, sir. It was very good lecture series, and I'm actually new to mycology, and I wanted to get to know what exactly kind of research takes place in the field of mycology. And it was a really enlightening and enriching experience attending this lecture series, and I thank you for giving me this opportunity to attend it, and I thank all of the people involved. They've done a very good job with the organization. Thank you.
Thank you, Mr. Shailesh, for your feedback and for your kind words. Thank you so much. Mr. Devashish Samant?
Hello, good afternoon, sir.
Good afternoon, Devashish. Please go ahead. Yeah.
Okay. Sir, it was very wonderful presentation by all of the faculties. Thank you, Belle sir, along with your all team. It was a wonderful session. I really enjoyed that moment. As it is very much new for me, but my suggestion, it will be more useful if such lectures on presentation on such medical field, for example, pathogenic organisms causing the pathogenic fungi which causes diseases in human beings. If you can organize by some such presentations in near future, so it will be more useful for us, sir.
Thank you, Devashish. We invited one of our associate editors from Sri Lanka, who is an expert in medical mycology, to give a talk. But because of some personal issues, she could not agree. But we'll be conducting, some events on medical mycology, because medical mycology is very, very related to human beings, health of the humans, welfare of the humans. We'll be doing such events in future. Thank you for your kind suggestion, Devashish.
Thank you, sir. Thank you, all of you.
Mr. Sanjay? Anyone? If I missed anybody? Okay, maybe.
Yes, sir.
Mr. Hamad, please.
Yeah, thank you, sir, for arranging such a good and fantastic presentations. So, as we hear from professors, doctors from different countries, so I request that in future, if you invite a scientist or professor and a doctor and lecturer of university from Afghanistan as well, to share the information and also their whatever they have collected in their area, and maybe there will be some new about the fungi, that I request in the future, if you invite a scientist in the future, such kind of presentations and presentation, it will be very best, and also for sharing information.
Thank you, Mr. Hamad. To be frank, this is the beginning of a joint event for MycoAsia. Thank you to Sri Venkateswara University, Tirupati, for believing in us and proposing, asking us to have this kind of meeting. We would be happy to have collaboration at the SARC level, full SARC level, including Chinese and Thailand and Indonesia in coming years. Be assured that we will be looking into that. We'll be working hard towards that. Thank you, Mr. Hamad.
Okay, thank you, sir.
Yeah.
Yes, sir. Thank you, sir, for organizing this workshop. All the sessions are very informative, especially the first technical session was very helpful for those who just starting the identification of and the nomenclature of fungi. Thank you, sir, for organizing this. And definitely we want more such a workshop in future. Thank you, sir.
Thank you, Pushpavati.
Thank you, Michael, and the whole MycoAsia team.
Thank you, Pushpavati, for your kind words. We will work hard in coming events to make it better and smarter. Thank you. Mr. Anuj?
Am I audible, sir?
Yes.
Yeah. Thank you so much to the whole MycoAsia team for organizing such an informative lecture series on this wonderful day of the World Fungi Day. And actually, I have to say that, in my opinion, I think online event has ensured the wider participation from all around the globe and across the, because most of the time the students and the scholars are not able to attend if this has been in the physical mode. So thank you so much to MycoAsia team for organizing it online. So hope to have more such informative sessions and lecture series in the future as well. As you have mentioned about the medical microbiology, so please be focused on their topics as well. Thank you so much.
Thank you, Mr. Anuj. I mean, we really appreciate the strengths of online platforms.
Yeah. Thank you, because
we really understand the strengths of online platforms, and we are leveraging it to the best of our capacity. So we are trying to people from all over the world, starting from SAARC region to China to Thailand, and Indonesia, Malaysia, maybe Japan, New Zealand, because today morning Dr. Bevan was there from Auckland. Okay, we will try to have more of online plus also some offline meetings also. In November, we will have an offline event in collaboration with Shri Venkateswara University. Okay, so it will be a mix of both online and offline in coming years. Just believe in us, keep supporting us. Thank you. I think Mr. Shazina will be the last person to actually give the feedback. Then we'll ask our co-founders to give their concluding remarks. Mr. Shazina. All right. Dr. Rajesh Kumar, your final remarks about the whole event in the future.
Thank you, the organizing committee from the university, and Dr. Dam for taking all the pain and efforts to make it a grand success. As far as the talks are concerned, it was very good in terms of content as well as the very topics we have covered. And I really looking forward for much more presentations and such events in near future for the benefit of mycology population. I wish everyone a warm World Fungus Day, and we are looking for more fungi and mycobuddies to join MycoAsia and contribute significantly to our team and journal. Thank you.
Thank you, Dr. Rajesh. Thank you very much. I mean, you gave a very, very informative talk on about the role of culture collection fungi in the morning. I mean, there are many things I learned from your today's talk. Thank you very much. Now I move to Professor Samantha Karunarathna. Do you have final remarks about the whole event, Professor Samantha?
Yeah, thank you very much, Dr. Shenoy and whole team for organizing this event. And it was indeed a wonderful session we had. And thank you very much for promoting mycology in Asia and whole world. Dr. Shenoy should be thanked for this effort he's taking to promote the mycology. And I also feel that we should have more workshops and talks like this in order to promote the mycology in Asia and whole world. So happy World Fungus Day, and thanks again for the organizers for inviting me for this event, and looking forward for more workshops and talks in the future. Thank you.
Thank you, Professor Sam. I mean, I foresee a future in which participants go to Sri Lanka for training on workshop under MycoAsia banner. Maybe from Sri Lanka to Pakistan or Afghanistan, or from Bhutan to India, or from Sri Lanka to maybe China or Thailand. So under MycoAsia banner, I mean, all your support will be very essential. All means all the mycophiles of the world. Your support will be very much needed then. Thank you, Professor Samantha. And now I request Professor Chinthala Paramageetham, head of the microbiology department at Shri Venkateshwara University, Tirupati, India, to give her concluding remarks about the whole event. Please, madam.
Thank you, sir. Good evening to all the participants and MycoAsia team. We have reached the end of this event. This event is an outstanding example for scintillating, exciting, thought-provoking talks. After listening all these talks, really our minds have been assailed by a torrent of ideas, information, interpretations, visions, and our own perspectives. This is really a time to meet the challenges in mycological studies and promote mycology research in universities as well as in research institutions. This seminar event organized really adjudged a success on mycological research challenges. I congratulate MycoAsia for designing intensive, high priority talks covering whole spectrum of fungal studies starting from taxonomy to industrial importance and medical importance, which assume high-level participation and interaction and discussions. I would briefly like to review some of the main points said during the deliberations. So the first talk by Nalin Wijayawardene throws light on taxonomic details and nomenclature. The second talk by Professor Samantha highlighted poisonous mushrooms and mushroom toxicity recognition based on multi-grain cascade system. Third talk by Professor Belle, Damodara, Shenoy on species versus species complex covered the role of polyphasic taxonomy on genealogy establishment. And the fourth talk by Rajesh Kumar KC give detailed information about various repositories, CBDs, culture collections, and preservation of fungi. In post-lunch session, first talk by Rohit Sharma covered bioremediation aspects of fungi. The talk by Fungi of Indian Ocean highlighted about the deep sea fungi and sampling strategies by Samantha Pai. The talk by Gunjan Sharma on transcriptomics of plant pathogenic fungi is very lucid and informative talk. The last talk by Shri Parimal on healing with mushrooms introduced entrepreneurship opportunities in mycology. I thank rapporteurs for making the session vibrant. My own feeling is that mycology research is to be encouraged and requires serious and urgent attention to turn young researchers into mycophiles. I am sure MycoAsia certainly acting on that. On behalf of our university, we are also interested in collaborating and spread the news. The postings in MycoAsia and by Dr. Damodara Shinoy postings impacts and encourages all mycophiles who are around the world. So, sir, in the Department of Microbiology, Shri Venkateshwara University, in future, we got this wonderful opportunity, and in future also we are very much interested to collaborate in conducting seminars, workshops, and even in international events also. We are looking forward to collaborate in future, sir. Thank you, sir.
Thank you, Professor Chinthala Paramageetham, madam. I mean, first of all, I would like to, thank you from the bottom of my heart, because thank you for believing in MycoAsia. You came to us and you wanted to collaborate with us, and it has been a wonderful opportunity to showcase to the world that what we are made up of, actually, what MycoAsia is made up of. Thank you again, and we will have more events with you. And thank you very much. And secondly, I would like to thank the participants of today's event. You have been really awesome. Without you, MycoAsia is nothing. Without you, any collaborative joint event is not going to happen. You have been awesome to MycoAsia team, MycoAsia initiatives, and I thank you very much for your kind support and encouragement. And we will be in touch with you. We'll keep interacting with you. We will try to understand your requirements, your aspirations. What are the things where we can play some role as a MycoAsia team, as a journal, as a group of mycologists from South Asia, extended to China and Thailand. Thank you very much again. And I would like to acknowledge the greatness of my MycoAsia team. It has been so wonderful to have such talented people in MycoAsia team. I also invited speakers like Mr. Parimal, Udgave. Thank you very much, the team members. You guys have made MycoAsia a proud entity in today's world. Thank you very much. And with this, I would like to conclude this event. Maybe we can leave the event now. Thank you so much.